Dear all, I just want to request you, please rename your entry in this Zoom. Actually, somebody is entered with the name admin. So please, I request you, please rename yourself. Soon, next speaker will join us.
अभी 2 मिनट हो जाए।
Madam, our speaker, Dr. Pagmalkar has joined.
करने से वही कि मतलब नहीं है, जैसे हो जाएगा तो फिर वही प्रॉब्लम है।
Ashish sir,
Yes, ma'am.
am I audible?
Yes, ma'am.
Okay. Good afternoon, everyone. I, Aparna Mandle, Assistant Professor at CDIP, welcomes you all in today's session, today's next session of AICTE-RGPV Joint Teachers Training Program on Artificial Intelligence in Pharmaceuticals, Contemporary Status and Impending Prospects. Without taking so much of time, we are going to start our next session on machine learning in drug discovery and development with Dr. Parimal Kar. Dr. Parimal sir is an assistant professor in IIT Indore. He is an alumni of IIT Bombay and Michigan Technological University, USA. He was visiting research associate at Michigan State University, USA from 2012 to 2016. He was also a guest scientist in Max Planck Institute of Colloids and Interfaces at Potsdam, Germany from 2010 to 2012. He was a visiting student and visiting scholar in Forschungen Jülich, Germany in 2007 and 2008. He was a graduate intern in Center for Nonlinear Studies, Los Alamos National Laboratory, USA. He has got many fellowships and awards in these years. He is a member of Biophysical Society and American Chemical Society. His area of research are structure and dynamics of proteins and nucleic acids via multi-scale, coarse-grained all-atom simulations, molecular dynamics simulations of glycans and protein glycans, complexes, multi-scale modeling of biomolecular recognitions, protein membrane interactions via molecular dynamic simulations. He has more than 20 publications in various international journals. With such an outstanding set of achievements, it's our honor to introduce you as our next speaker. Dr. Parimal, sir, over
Yeah,
to you.
thank you, ma'am, for such a nice introduction. So I would really like to thank the organizer for giving me an opportunity to talk to in this program. So I don't want to take much time because I have only 1 hour time. So I'm gonna switch up my video because since I'm traveling actually, so my bandwidth may not be that good. Okay, so I'm just switching up my video. I'm just keeping. Uh, so can you see my slide?
Yes, sir. It's visible.
Okay. Okay. Thank you.
Yes, visible.
Yeah. So, so today I'm going to talk about computer-aided drug discovery. Okay, mainly, basically, it's applications of machine learning in drug discovery. Okay, and the talk is organized accordingly. Like first, I'm going to talk about molecular docking, and then give you an introduction to two techniques, unsupervised machine learning techniques, that is k-means clustering and principal component analysis. And then we'll talk about large-scale MD simulations, and we'll show how these two algorithms, these unsupervised algorithms, are used to study the protein dynamics, and also in the context of protein-drug interactions. Actually, I'm going to mostly talk about that way. Okay, and applications part, basically all this example would be mainly from SARS-CoV-2. Okay, so that's what I've been doing in last 7-8 months work that I'm planning to show you. So just to start with molecular docking part, you know that simply put, docking attempts to find the best matching between two molecules. Okay, so you have the target protein, that is the receptor, and you have the ligand, that is your drug. So basically, since inhibitors are small molecule, they are very flexible. Okay, so what you need to find basically at the binding site of the protein, what is the best pose that implies to that generates maximum interaction with the receptor protein. At the same time, it minimizes the total energy. Okay, so the best pose refers to an orientation where the number of interaction is maximized, thereby the total energy is minimized. Okay, so that's the thing. So now when we try to do all this things talking, but we need to see actually how here also we can use machine learning things. Okay, so for example, let's talk about these two drugs, okay, aspirin and sildenafil. So as a chemist, you see these two drugs, okay, like these atoms, and then single bond, double bond, this ring structure, everything you see like that. Okay, so now, can I use it for machine learning purposes? No. If you have this kind of structure, then you cannot use it for machine learning things. So you can convert this into this thing called SMILES, okay? Simplified Molecular Line Entry System. So if you just convert it into that kind of way, so then now you see that all these 2D figure that you have, if you just convert them into this linear entry system, then you can use any machine learning algorithm such as natural language processing. Okay, you can use any NLTK tools, natural language processing tools, to study all these systems. Okay, now this is how a chemist see small molecules. But the question is, how does the biology sees? How does the protein see these foreign molecules, okay, that they wanted to bind? So they see instead, actually, the proteins actually, they see aspirin and sildenafil like this, okay? Here, basically, it's a electric field. They look at it like that, okay? It's a van der Waals surface, and on this surface, as if, because since it composed of many atoms and each atom has charge, so that means there is some electrostatic potentials, okay? So so you just project this electrostatic potential on the surface, this van der Waals surface, okay? So that's how it looks like, and the protein molecule sees these molecules, okay, like this kind of things. And then also you also need to look at, I mean, how does the protein binding site looks like, okay? So that also looks like this, okay? We see it in terms of hydrophobic or hydrophilic surface. Now, this is very important to design a drug, okay? Once you can characterize your binding site, okay, whether it is hydrophobic or hydrophilic accordingly, or what kind of charges are present in the binding site, then you can accordingly modify your initial molecules and make it a better drug candidate, actually, okay? So this is one such applications that, I mean, in molecular talking, when we do it in the drug discovery process, we should keep this thing in mind, okay? And hello?
You are audible, sir.
I'm audible?
Yes,
Yes,
sir.
sir.
But unfortunately, why my laptop is stuck? Because I cannot, it got stuck actually. Back. Okay. Okay, this is okay. So now this is how we go ahead with this drug discovery things, and this is very important, as I said, because this in the drug discovery's importance is like you can develop drug, okay, like inhibitors, antagonists, etc., that block the recognitions, or uh, this thing called increase or emulate the recognition process, that's called agonist or recombinant protein, etc. Okay, so to understand all these protein drug interactions via molecular talking, okay, that helps you to design new drug molecules. Okay, so overall, molecular docking refers to the process that involves placing molecules in appropriate configuration to interact with a receptor and form a stable complex with a minimum energy. Okay, so this is a natural process that always occurs within second in a cell, but when you want to simulate this in computers, you cannot do it in a second. Actually, it takes a lot more time, okay, because it can have multiple orientations in the binding site, and you have to evaluate the energy for each orientation and then compare and then find, okay, for which case this is the minimum energy, okay. So now here I show that, I mean, this blue one is the protein, and the binding site is shown in a red curve, and the ligand is shown is green. So your job is just to place this ligand molecule inside the binding site with an orientation. As I said, the orientation should be the ideal orientation, should correspond to the maximum interactions and the lowest energy. Why it is lowest? Because the total energy is always negative. Okay, so that's the binding free energy. Basically, the binding free energy that you calculate is called the Gibbs free energy, actually. Okay, so the Gibbs free energy should be negative. So more the negative is better, the stable the structure is. Okay, so that's how we go ahead with these things. And Next, basically aim of this molecular docking is to achieve an optimized conformations and relative orientation between protein and ligand such that delta G, that is the Gibbs energy, is minimized. And you call it in the medicinal chemist or in pharmaceutical industry, this delta G, we call it the binding energy or binding affinity. Okay, but in physics, this is called the Gibbs energy. That's what basically in thermodynamics we call it. And this is to study whether two molecules interact with each other, and if so, what is the orientation that maximizes the interaction, which minimizes the total energy. Okay, and the goal is to given a protein structure, predict its ligand binding. So that is the job generally we do it. And we do it by virtual screening because we have a large data set. So for example, the current database, maybe we have around 1.6 billion of compound. I mean, I worked with 1.3 billion of compound that I did the virtual screening. Okay, we published this paper also. So yeah, this identifying drug candidates by considering large number of possible ligands, and then lead optimization. Basically, you need to modify the drug candidate to improve its drug likeness property. Okay, and then if the binding pose is of the candidate is unknown, then docking can help you. Okay, you can generate that docking pose, and docking can also predict binding strength of related compounds. Okay, so but the problem is that the initial drug candidates are often bad drugs. For example, although they might bind to the target, they often don't bind with high enough affinity. So then the question is, how can you make it a better drug candidate? Okay, so we do this lead optimization. We can perform molecular dynamic simulation, get some insight from it, and then we can optimize. I mean, we can add or delete some portion of the molecule so that they can form new contacts, van der Waals contacts, or electrostatic or the hydrogen bond contacts with the receptor protein. Thereby, they can have a better binding affinity. Okay, so that is we want to do basically. So, but there are different ways to do all these docking things. The models, I mean, this is a pretty old method. You know, in 1890, there was this traditional lock and key method. It was by Emil Fischer. This is very, you know, that you have a lock and you have key. So you know that only one key can open that particular lock. So it is like that. But the problem right now, the thing is that this traditional lock and key model may not work for say. So these days we are working on carbohydrate-based drug molecule. Okay, now there lies a problem because carbohydrates are very flexible molecule, and the other thing is that since they are very flexible, so at room temperature they can have multiple conformations. So that means that in the case of glycan-based drug, you have a lock, but you have bunch of keys, and each key. So these key basically are same protein, I mean same glycan, but they have multiple conformations. And this key, so you have a bunch of keys, and each key can bind to different protein. So each key can open different lock. So it's an entirely different ball game actually. You know, it's very difficult now when you want to design carbohydrate-based drug molecules. So their flexibility plays a role for which you need to study the dynamics of the ligand molecule, okay? That is the glycan molecules. And then you have to predict which one is the most probable conformations at room temperature, and then you can design then and need to check that whether that particular conformations bind with the target protein or not, okay? So there lies the importance of doing molecular dynamics study for this kind of designing drugs. So that was 1890, and then 1958 we had the induced fit model, okay? That's by Danny and Cosland. That's actually a better method because you know that often when the protein and drug binds, they may not be rigid. The binding pocket has a different shape and the ligand molecule has a different shape, but when they come too close to each other and try to bind, both of them adapt their structures, okay? They basically have some conformational changes, and so that they can form a tight binding with each other, okay? Then 2003 came the conformation ensemble. So that is even better method. But remember one thing, that all models are wrong. Some are useful. Okay, this is by George Box. Why all models are wrong? Because when you say modeling, it means that you are making some approximations. Now, your approximation may work for that particular cases. Okay, it may not work from some other cases. So molecular modeling in drug discovery, okay, or any kind of modeling. So this is just an approximation of the real problem. Okay. In modeling, we always try to make a balance between speed and accuracy. You can improve the accuracy of your model, but at the cost of what? At the cost of speed. If you have a large supercomputing facility available, okay, go ahead, use the best method available with your hand. You can use that one. But in today's drug discovery scenario, I mean, for example, this COVID-19 cases, okay, we don't have much time. We need a drug very urgently. So why do you have that much time? Okay, so for all these cases, actually, we need to develop new methods so that that method can enable us to make a fine balance between speed and accuracy. That is the main purpose of any kind of molecular modeling, whether you use AI or machine learning. All these techniques are developed only to facilitate that thing, to make a fine balance between speed and accuracy. That's all. You have 1.3 billion compounds. You can screen all the compounds one by one. Okay, but then how much time it's going to take? So for that one, you have to use the machine learning techniques to screen, to do the virtual screening for 1.3 billion compound. Okay, so that kind of challenges today we are facing. We don't have much time. We have to get the result very urgent. I mean, quite fast. Okay, so that's why all these different kinds of methods are being developed. So I already talked about this method actually. Okay, so this is key unlock. You have this key, the protein and the receptor both are very rigid. They basically snarkily fitted to each other, just like key and lock. And this one is the induced fit. As I said, that in this case there is a conformational changes occurred. Both basically, both the ligand and receptor both adapt so that they can have a maximum interactions between them. The third one is this conformational ensemble approach. Okay, so this is also basically the energy dictates. You have pre-existing ensemble of conformational state, and then basically this is good because you take into account the plasticity of the protein that you take into account in this case, and then you try to just document which protein conformations the binding is, gives you the lowest free energy. Lowest means more negative, most negative binding free energy that gives, and that would be your the protein ligand complex structure. Okay, so now the problem that we face in today is that the main bottleneck is that we don't have actually often we don't know the three-dimensional crystal structure of the target protein. That is one of the biggest bottleneck that we are facing. So now, for these purposes, actually, we use a lot of different methods to generate the 3D structure of the protein form sequence. For example, I don't know whether you guys have read the paper or not. Google has a platform called DeepMind. Okay, so,
paper.
yeah. So that paper that Google this is called AlphaFold. So they use deep neural network methods to predict the three-dimensional structure of a protein from its sequence. And basically, last 50 years we could not did much actually. But when they use this deep learning method, now the accuracy level is pretty high, and they did excellent thing. And this code is freely available. Actually, you can download and you can use it. So they now developed, I think, AlphaFold version 2. Okay, so that's where you see that application of these different machine learning approaches or artificial intelligence in predicting the three-dimensional structure of proteins also, while we don't know the structure. So, for example, when this SARS-CoV outbroke, okay, we didn't know that three-dimensional crystal structure of these proteins, these 3-cell proteins. Later on, we got the structure that is published in Science, but initially it was not available. So in those cases, actually, this kind of method is pretty good. The main problem with any other methods, those are also they use some extent machine learning technique to predict the three-dimensional structure. So it is very easy to predict the this secondary structure because those are rigid. You can predict it. Main problem is the loop modeling, and you know that often the dynamics of loop plays a role in the function. Okay, so basically structure determines the function. So if you can predict the loop structure properly, then it would be very difficult for you to develop a drug targeting that protein. Okay, so the accuracy level for predicting the structure should be high, so that when you use that structure instead of the of structure, that would be. Any question? So that can be used for your drug discovery purposes. Okay. So if you have not if you have not read that paper, so that paper is actually freely available. Okay, you can download it from Nature. It is called AlphaFold. That software. It's Google's DeepMind lab. From that lab, they have done it. So they use this deep neural network method for that one. Anyway, so generally this docking, you have a scoring function and then there is a search method. Okay, so those are roughly, scoring function means that roughly approximate the binding affinity of a ligand to a protein. Okay, basically what are the different interaction between the protein and ligand can have. So that is the scoring function. And the second one is the search method that searches for the best scoring binding post. That means what are the different orientations a small molecule can have in the binding pocket. So these are the two different parts. So post generation is, so the searching method means the post generation. So that is a solved problem. Okay, we can do that thing. We have a lot of different methods that we can do. So currently the main focus, I mean the active research is still going on in the scoring function. Okay, you have generated a pose, okay, orientation, but what is the binding energy for that particular orientation? How accurately you can predict that binding energy? So that is called the scoring function, and still there is a lot of room available to work on that area. Okay, another bottleneck is that generally we treat protein as a rigid molecule and then ligand as a flexible molecule, or both we can use as a rigid, or both can be flexible. Okay, but generally the freely available softwares that you use, for example, Autodoc Vina that you often use, so generally there the protein is rigid and the ligand is flexible. Okay, so it's a semi-flexible kind of method, but you know that in reality proteins and ligands both are flexible. Okay, and water molecules also play a major role in determining the binding affinity, but in Autodoc Vina you cannot use those water molecules. Okay, so that's why you always need to basically complement your docking study with molecular dynamic simulations, where you take into account the dynamic aspect as well as the solvent aspect both. Okay, so well, I just told that there are three different kinds of docking: rigid, and then semi-flexible, and the flexible receptor. The problem is that when you use that both receptor and ligand are rigid, okay, so then it is a very fast and simple method. Then the most commonly used method is receptor rigid and the ligand flexible. That is a semi-flexible method. That is a fine balance between speed and accuracy. Okay, when both are flexible, that is very slow and complex. Okay, so if you have a very large number of molecules that you want to dock, then of course this may not be a good idea. Okay, the flexibility things may not be a good idea. So you see that constantly there is always this balancing thing that we do, okay, between speed and accuracy. That's what we do all the time in molecular modeling things. And this figure actually shows that how the computational time increases significantly when you move from the rigid body docking to flexibility, fully flexible, where you lay the backbone and the side chain both flexible. Generally, some softwares are available that allows you to make the protein also flexible, okay, as well as the ligand. But they allow the side chain only to be flexible, not the backbone. Backbone flexibility is not that easy. Okay, so if you do that thing, you can see that how many CPU times increases so rapidly. This is just exponential increase. Okay, so that is the challenge that we are facing in molecular modeling in drug discovery. Well, so there are three components. When you use any docking software, it has three different components. Okay, first one, how do you represent the molecules? Okay, it can be atomistic representation, or it can be, say, Corskin approach, or it can be united atoms. And then the second one is the scoring function. As I said, in this area, still active research is going on. Okay, so it is used to assess the quality of the dock complex. Okay, how do they do it? By computing the total binding energy. And the third one is the searching algorithm. You have to exhaustively search all these different conformations possible, or a ligand can adopt in the binding pocket. Okay, so you can have exhaustive sum, Monte Carlo, genetic algorithm, simulated annealing, taboo search, all different methods are available that you can use it. So now the question is, once you generate the conformations, then what next? Okay, so now the post generation, that is the place the ligand in the binding site, and this is a well-solved problem I told you. And then post selections that determine the proper pose. This is the hardest part. The pose is selected by using the scoring function. Okay, while active research is going on. And so that's why what we do basically, we use the multi-stage approaches. As I told you that since you have 1.3 billion compounds that you want to screen, how would you do that one? Okay, generally, if you have thousands of 1 lakh molecule, you can use this kind of algorithm that you can use. Say, for example, you are starting with 2 into 10 to the power 6 compound. Okay, and if you have access to 100 CPUs, then use glide HTBS. So that takes almost 2.3 days. Okay, so one compound takes 10 seconds. So now, if you use glide standard protocol, this is sorting the software suite. Okay, so here you see that, so this is a slightly better, they use a slightly better scoring function. Okay, so suddenly there is a 12-fold increase in the computational time. Now for each compound you need 120 seconds. Okay, now when you go for the best model, the best scoring function that you use, the glide XP, okay, extra precision model, then it's even further. Now for each compound, now you take 10 minutes. Okay, so from 10 seconds to 10 minutes. So that's the thing. So you cannot use this method for the initial screening. So we use generally all these multi-stage approaches to reduce the number of molecules in each stages. And finally, we do this visual analysis, further refinement, synthesis considerations. Okay, we do, then with whatever you get, then you can do toxicity analysis. Okay, ADMET, basically admit properties you can analyze, thereby further you reduce the number, and then you perform MD simulations, and thereby you can reduce the number further down to 5 to 10 molecules. And then you can say, okay, these are the 10 molecules, you can give it to the medicinal chemist. Okay, you can synthesize it, and then they can go ahead for experimental testing. Okay, so that's how we can complement the experimental study using various computational approaches. Okay, so now another thing that I want to tell you in molecular docking that is used in drug discovery is this. When you do docking, generally we have a tendency to take the complex structure for which the total energy is the minimum. Okay, that can sometimes be misleading. For example, if you consider this case, okay, so the lowest energy that you find in the first cases, okay, so you thought that maybe that is the best pose. But the real side corresponds to the second one, okay? I just pointed out with real side. Why? Because for the first one, the well, okay, this potential well, that barrier height is 10 kcal per mole. For the second one, although the total energy is slightly higher than, okay, so that means slightly less negative compared to the lowest energy, but the depth of the potential well is much higher. It is 15 kcal per mole. So that means when when the ligand is bound in these conformations, the barrier height is so high, it cannot come out. So it should be a tighter band, real binding site. Now, you don't know actually whether this one is the best one or that one is the best one, because you cannot measure the barrier height. So how do you know? Then what would you do in that cases? Okay, then we can do the molecular dynamic simulations. Over the time, if this site is not the real binding site, you can expect that if you run a long time MD simulations, the ligand should come out from this lowest energy structure from which you have started the simulations, and then it should come to the real binding site. It will readjust itself and reach to that binding conformations. Okay, and then you should evaluate the binding energy again, and then you can say, okay, that is not the real binding energy, but this is the real binding energy. So if you then basically based on this, if you predict that, okay, this is the best compound I have found, so it could be then misleading. Okay, so these are the different pitfalls out there that you should take into considerations when you do the drug development using docking study. Okay, so these things you all know, that this rule of Lipinski's rule of 5 and everything that you know, I don't need to talk about it. But one thing that I just want to talk you, that I always say that, this molecular modeling is always in speed and accuracy. So docking is more accurate than the virtual screening. But docking at the same time, you cannot use large number of molecules for molecular docking study. If you have large number of molecules, then you should use the virtual screening. Okay, but it is least accurate. That's the thing. Okay, so you should always keep that in mind. So now I told you that sometimes, often, you can get stuck in a position which is not the real binding site. But since molecular docking is a static thing, you cannot come out of that, from that position, and go to the real binding site. Okay. So for that one, we should, and also in molecular docking, you are not taking into consideration the effect of solvent, that's water, and often there could be a water mediated hydrogen bonding also. So to complement this molecular docking study, as well as the Excel crystallographic study also, okay, once you solve the Excel crystallographic structure of the protein and the ligand, still you know that Excel crystallography, you do it in a buffer solution, not the real biological solvent. Okay, and also Excel crystallography is a snapshot, it's not the dynamics part. I know that it is the dynamics that controls the function. Okay, so that's why we need to complement this experimental or molecular docking study by performing molecular dynamic simulations. So using molecular dynamic simulation, you can calculate the free energy, that is the binding free energy. Not only that, you can decompose the binding free energy into different components, okay, different forces. What is the contribution of each forces to the total binding free energy, that you can compute, that otherwise you cannot get it from experimental study. Okay, so this is very important, because once you know that, not only that, you can then calculate what are the different amino acids that are contributing most to the binding free energy and which are contributing least in the vicinity of the binding site. Then of course you know that, okay, if I add one more maybe functional group to this ligand, maybe it can maximize the interaction with those amino acids in the binding cavity which are contributing less. Okay, so that's how you can optimize your starting molecule, and you can have a better drug molecule. Okay, so there are different statistical mechanical methods those are available that you can use it to study the compute this binding energy. For example, energy perturbation or thermodynamic integration, that is the most accurate method, but at the same time, you need a large computational resources to do all these calculations. On the other hand, I told you that docking is first method, but it's the least accurate method. Okay, so there is another called LI method, okay, linear interaction energy method. That is also very fast, but not that accurate. So we use a compromise between speed and accuracy, and we use, say, MMPBSA method, okay, where MM stands for molecular mechanics, PB stands for Poisson's-Boltzmann, and SA stands for surface area. Okay, we solve this differential. Generally, here we solve this linear. Basically, Poisson's-Boltzmann is a nonlinear partial differential equations. Now, the problem is with nonlinear partial differential equation is that it is numerically unstable. Okay, it's very hard to solve these equations. So we what do you do basically? We again we do modifications and make it a linearized partial differential equation, and then we solve it because that is numerically more stable things. So this is the method. I don't want to talk about this thing. Okay, this is the theoretical foundation of this method that's beyond the scope. Molecular dynamic simulation, we perform it, as I said, to complement the experiment or molecular modeling study. So here, what do you do? So we solve this Newton's second law of motions, okay? So that you have studied in your +2 level, or maybe in 10th standard, actually, you have studied this basic physics thing. So force is equal to mass time acceleration, and force is gradient of potential energy, okay? So that is, if you take a derivative, negative derivative of potential energy, you will get force. So what you essentially get are second order differential equations, okay? And you have to find position that is r as a function of time. What do you need to do? You need to just make an integration of this, double integration, then you should get it. But the problem is that the real challenge is that this is a coupled differential equation. For example, you want to study, say, spike protein in solvated water molecules, okay? You will have almost, say, 1 million atoms, including water molecules, okay? So if you have 1 million atoms, okay, then not 1 million, you will have something around 3 lakh atoms actually. So if you have 3 lakh atoms, then you will have 9 lakh degrees of freedom. So that means in MD simulation for that spike protein, you have to solve 9 lakh coupled this second order differential equation. So that is the challenge. And we have to solve it simultaneously and for a long time scale. Okay. So these are the different challenges that we face in to studying the protein dynamics. So generally, this the potential energy, this V, that is, we have to take the derivative of it. So it has all these 5 different components. First one is the van der Waals interactions. Okay, second one is the electrostatic interactions, that is the Coulomb interactions, and the third one is the bond stretching. Okay, so this is harmonic potential. Fourth one is the angle stretching, that is also harmonic, and the fifth one is the dihedral angle. Okay, so that is also harmonic in nature. So these are the 5 different components that we use to model the potential energy, and you have to take a gradient of it, okay, derivative of it for each atom, because force is acting on each atom. Okay, so that is the challenge. You see that when you use the software, you are just getting the result, but if you deep look into the method that they use, okay, so you can see that how much complexity is there to solve all these things. Anyway, so generally we use Barley algorithm to solve these equations. Okay, so that is very fast method, or you can use velocity Barley algorithm. You know, different other methods also, like RK method, you know, or the trapezoid rule. I mean, those who are in computational guy, they know all this method, but those are not suitable for this kind of study. Okay, because those are really unstable thing. So we use either Bartlett or velocity Bartlett algorithm. Okay, so now that's what it is. Different things. Now, what are the different machine learning things that we use? Okay, so after getting, so as I say that I show only two different algorithms. One is the k-means algorithm, and the other one is the principal component analysis that we use. Okay, so k-means clustering algorithm that was proposed by Hartigan and Wong in 1979. So it's pretty old. Okay, so almost 50 years old. 40 years old method. So here you have n distinct objects, and you want k-means clustering algorithm, basically that partitions the object into k number of clusters. Okay, such that cluster similarity is high, but the intercluster similarity is low. Okay, so here we have to specify the number k. K is the number that we need to specify how many cluster that we want. Okay, so that's the number of cluster, and consider the object are defined with numeric attributes. Okay, so thus using any one of the distance metric to demarcate the cluster. So that's what you do. Okay, so it's very simple algorithm. So first it selects k number of objects at random from the set of n objects, and these k objects are then treated as centroid or center of gravity of k clusters. Okay, and then you iterate it. Basically for each of these remaining objects, it is assigned to one of the closest centroid. So that means then it will form a collection of objects assigned to each centroid and is called a cluster. Next, the centroid of each cluster is then updated. So it's an basically iterative method. Okay, so this assignment and update procedure goes on until it reaches some stopping criteria, such as number of iteration or centroid remains unchanged or no assignment, basically that. So before cluster you have this, and after the k-means cluster you have these three different classes that you will have it. Okay, so that is one thing. And another one that is in biology actually that we face in drug discovery also. So when we perform MD simulations, okay, so we need to analyze the data. Okay, so now I said that to if you solvate the spike protein of SARS-CoV-2, you will have say 3 lakh atoms total. Okay, 3 lakh atom means 9 lakh degrees of freedom. So now in MD simulations, actually what you have basically the total dimension would be 6n. So total dimension for this, if you have 3 lakh atoms, you will have 18 lakh dimensions. Okay, 6n dimensions. So 3 coordinates, okay, x, y, z coordinate, position coordinates, and 3 momentum coordinates, px, py, pz. Okay, momentum because velocity means momentum. Okay, because m into v is momentum. So now what do you do? This is such huge dimensions. How can you make sense of it? Okay, you have to make sense of this data. You cannot visualize it. You cannot make sense of such a high dimensional data. But that exactly you are generating in MD simulations for protein drug systems. Okay, now to make it simple, I give you an example. So for example, you work in a pathological lab and you have 53 blood and urine measurement from 65 individuals. Okay, so forget about those such high dimension. Now you have only 53 blood and urine measurement and 65 individuals. Can you visualize the measurement? You can say, well, why not? I'll make a matrix, 65 by 53 matrix. Okay, I can do that. Well, you can make it, but it's difficult to see the correlations between the features. Can you make any correlations? Of course, no. By looking at this, you cannot. Then you say, well, I can. What I do, I'll make 65 pictures, one for each person, because you have the data for 65 persons, okay? Well, you can make, again, can you make any correlations? No. Then you said, okay, instead of 65, I'll make 53 pictures, one for each feature. You can make it. Can you make correlations again, looking at 53 pictures? No, you cannot. Okay, so then these problems are there. Okay, so that's why we want to Basically, the question is there a better representation than the coordinate axis? Can we visualize the data in a smaller dimension, reduce the dimension, but without losing any information? Is it possible? Think about it. We want to reduce the dimension, but we don't want to lose any information. Is it possible? So it is possible. For that one, we use principal component analysis. Principal component analysis is another unsupervised machine learning algorithm. K-means clustering is also an unsupervised machine learning algorithm, and PCA is also an unsupervised machine learning algorithm. Okay, I don't want to go deep into these mathematical formulations because I'm running out of time. So basically here, what you need to do, you have to construct first you calculate the sample mean, okay? Then you subtract the sample mean, then you compute the covariance matrix, okay? The main thing is you have to calculate the covariance matrix, and then you diagonalize it. You diagonalize the covariance matrix. Then what did you get? You will get eigenvalues and eigenvectors, okay? You will get eigenvalues and eigenvectors. Eigenvectors shows the direction, and eigenvalues gives the fluctuations in that direction. That is the variance in that directions. Okay, you will get multiple principal components. Okay, so each vector is called the principal component. So you can have multiple principal component, but the question is it worth to look all the principal components? Actually not. Only first two components are good enough. Okay, so first two components means in this figure, okay, you have this, basically the U1, so that is the first component, and then the U2, that shows the variation, okay, the mean square deviations basically, okay, variance, eigenvalues basically. So these two is good enough, this PC1 and PC2, because for these two will shows the maximum variance present in your data set. Okay, so now you see that we have a very high dimensional data set, okay, and then we represent everything in the two-dimensional point. Now it is easier for us to visualize any two-dimensional data point. Okay, now why only two components we use? Because those two components shows the largest deviations, okay, largest eigenvalues they show. That means they are more important eigenvectors. So that's why we only consider the first two one. You can, if you don't believe, then you can also plot, say, PCA1 versus PCA2, then you can plot PCA1 versus PC3 also. You can plot that thing, or PCA2 versus PC3 also you can plot it, okay? You can check that way, and you can see whether that really makes sense or not. Maybe you will see that only PC1 versus PC2, these two-dimensional plot is good enough to describe the largest motion or the largest variation present in your system. In the context of protein dynamics, we talk about the motion, okay? Because we are into dark discovery, because same thing is used in other purposes also in machine learning. So here for protein dynamics, basically, we'll show the most dynamicity in that directions, okay? So this is the different, okay, I talk about now all these different methods. Now I'll show you the applications, okay? So first applications I will talk about is this, this is a 3-cell protease of SARS-CoV-2, okay? This COVID-19. So there are two inhibitors, B and C, okay? So the B1 is called alpha-ketoamide 13B, and efficacy corresponds to Z31792168. Okay, so that's another small drug. Now let us see. I mean, complement the experimental study. Okay, we have the crystal structure available. So what I did, basically, we perform 100 nanosecond MB simulations, and first thing we check whether the simulation have converged or not. Okay, so for that one we calculate root mean square deviation. This statistical component analysis we do, and then we plot it as a function of time and shown in figure A. Okay, you can see that for APO or hollow, both hollow complexes, they have reached to an equilibrium because there is no change after 60 nanosecond. Okay, last 40 nanosecond, it is almost constant. Then we look into the dynamics of the ligand. I told you that ligand are small molecule, so they are gonna be flexible. Okay, so what you want really actually that the ligand should snugly fit into the protein. If it is too dynamic, you may not have a very good drug candidate. So here you see that alpha ketoamide 13B from the crystal structure actually it deviates. So what they predict in the crystal structure is not the correct structure, correct orientation. It deviates and then gets stabilized at a value of 2.5 angstrom. Okay, so you see that it is characterized by a single peak. If you look at the green curve distribution, okay, it is characterized by a single peak and then there is a broad distribution. On the other hand, the smaller that shows bimodal distributions, and both are possible. And I told you that if it is like that kind of flexible, then it can't be a good drug candidate. Okay, so the green should be a better drug candidate than the blue one. Okay, so now this RMSD distribution shows like they are very flexible in the binding cavity. Okay, now you can do the clustering analysis. You have this 100,000, okay, 1 lakh conformations. Okay, you have the data set comprises of 1 lakh structures. Now you perform k-means clustering of this. So what you see that basically this is the two different classes that you get, and their percentage of occupations are also I have shown it here. Okay. Next thing that we study in drug in this. So what happens when a protein, when a protein is attached to a drug molecule or ligand? What happens to its atomic motions? So we calculate the correlation. Okay, but this is called DCCM, dynamic cross correlation. We calculated using this equation. Okay, so correlation is also another statistical method that is frequently used in machine learning. Okay, so here we apply this technique to study the impact of ligand on the dynamic motion of the atom. What do you see here if you look at the figure A? Okay, just look at the circle lesion R1. Okay, so here, well, the correlation coefficient that varies from 1 to -1. So +1 means it's highly correlated, and -1 means anti-correlated motions. Okay, and 0 means there is no correlation among them. So what do you see that here for R1, when the drug is not bound to the protein, you see that there are a lot of brown patches up there. So that means there is a lot of correlated motions up there. Now look at B and C. On that particular region, actually, you see a lot of yellowish thing. So that is also correlated motions, but it is decreased because the brown one is more stronger correlations. So after the drug binding in that particular region, the motion has become less correlated. Okay. Now look at the R2 region. In R2 region, in figure A, it is bluish. So that means those are anti-correlated motion. Now look at in B, that region has become red. In C also it has become red. So that is also anti-correlated, but it is stronger anti-correlated motion. So the drug binding enhances the anti-correlated motions in that particular region. Okay. Similarly, in R3 region, also the same case. It is increases. So overall, you can say that after the drug binding, atoms, basically the residual motions have become more anti-correlated. Okay, so that is one thing that you can say. Now I told you another application that I want to show you is the principal component analysis here. Okay, So here we plot PC1 and PC2, these first two principal components, in terms of energy. Okay, we convert it into energy using Boltzmann formula. Okay, minus RT ln PC1 divided by PC2. Okay, so that will give you this kind of energetic plot, and then you plot it, this two-dimensional contour plot. You can make it. Okay, so these all plots are made using Python. So now you see that for figure A, that is the APO structure, you see two minimas. You see, okay, the dark blue, that corresponds to minima, the global minimum, okay, and their contribution is 60%, and then in those structure you then again perform k-means clustering. So then you assign the number. So after doing PC, we again perform k-means clustering, and then we say that for these two minimas are there, and one minima has 67% confirmations and the other one has 33%. So then the most probable confirmation will correspond to that 67, okay, and the other one is the secondary one. Now, when alpha ketoamide 13B, which is a relatively larger drug molecule, when it binds, then you see that there is only a single minima. And if you do the cluster analysis, you see only 100% success belong to that one. So what does that mean? It means that the binding of this drug stabilizes the protein conformations. Okay, on the other hand, when the second drug binds, again you see two different minima. And one has 61% and another one has 39%. So again, this drug binding cannot enhance the stability of the protein. Okay, rather it has again flexible nature of the protein is there. So then you can say that alpha ketoamide 13B is a better drug candidate than the other one. It has to be. Okay. Now, when you do the principal component analysis, then we want to see the motion, in which part of the protein you see the largest motion, because I told you that because this principal component analysis, the eigenvectors will give you the direction and the eigenvalues give you the fluctuations. Okay. So now here the eigenvectors are shown. Just look at on the right hand panel pictures. Okay. So look at this porcupine plot. The green spike, those are shown. So those are the eigenvectors. So you see that in this entire protein, in which direction the proteins are moving, the amino acids are moving. Okay, and the length of that porcupine or length of that spike determines the fluctuations, how much it is fluctuating. Okay, so you can just look at ABC, figure ABC, then you can compare B and C with A, then you can see the impact of binding of drug on the direction of motion. Okay, how it is changing. Okay, so these are the different approaches. The application of various unsupervised machine learning algorithm to understand the protein dynamics. Okay. Do I have time or I have to stop? Excuse me? Hello?
Yes, sir.
Yeah, should I continue or I should stop because it's 12?
Yes, sir, you can continue.
Okay. So these are one applications in SARS-CoV-2. Okay.
Excuse me, sir.
Yeah.
Actually, it is a time to binding of program.
Yeah. That's why I asked whether I should.
I think this might be targeted, I think. And we will give you the 5 minutes for question hour sessions.
Okay. So yeah. So these are basically one thing I showed you, these applications of this clustering and different cross correlation is another machine learning techniques, and then principal component that I have shown you that we use to study all these various aspects. So principal component also has another variant that we use it in the context of studying the dynamics. Okay, so often what happened, I told you that the main problem is to model the study the dynamics of the loop regions. Okay, because they are very flexible. Why they are flexible? Because they are solvent exposed. Okay, so they can have multiple conformations. So we should do the principal component analysis for that particular region also to understand what are the different conformations that a loop can adopt in solution in the ligand bound condition, and what are the probabilities of different conformations. Okay, so now what we have noticed that if we use Cartesian coordinates, okay, like XYZ coordinates to study the dynamics of the loop region, it is not a good thing, because you know that the best way to characterize the conformational change in protein is to study the phi psi angle, that is the dihedral angle. So there is a new variant of principal component analysis available that is called dihedral principal component analysis. Okay, so where the collective variable, okay, or the reaction coordinate is chosen as these dihedral angles instead of the Cartesian coordinate. So the first one, the principal component I showed you, I choose XYZ coordinate of C alpha atoms. Okay, so here we are taking the dihedral angle. So if you do that thing, then you can have a more finer, that is, if we take internal coordinate as a collective variable, then we can better assess different conformations. Okay, otherwise, if you do Cartesian coordinate, use Cartesian coordinates and do the principal component analysis, you may not get the different variations a loop can adopt. Okay, so this shows again, basically the first one corresponds to the APO structure and the B and C corresponds to the protein bound structure. Okay, so what are the different loop, this connecting loop that connects this N loop and C loop of the SARS-CoV-2 3-cell protease. So there are different conformations that you can study using this method. Okay, so I don't want to talk much. Actually, I want to stop here. I have a lot of things to talk, but I know that only 1 hour time, so I don't want to take time your, but I'll stop here. Okay, so but before I stop, I just want to thanks my student. Okay, Nisha, Fulbabu, Rajeshri, Sayon, I would like to thank them because most of the works have been done by them. Then also my collaborator from my Susobon from my Palakka, Dr. Devasi. And also I have been supported, my group is supported by Department of Biotechnology and Department of Science and Technology. And also I would like to thank IIT Indore also for giving me startup fund. So thank you all with this. And if you have any questions, you can ask me.
Thank you so much, sir. If anyone is having any query or question, they can write in the chat box and we will discuss it. Anybody having any question or query, they can write in the chat box.
Okay. If anybody having any questions, just write down in the chat box.
Okay, thank you so much, sir, for giving the concepts of molecular docking and simulation. And it definitely helped the pharma people a lot. Thank you so much, sir.
Thank you. Thank you.
Now, moving towards our next session on hands-on artificial intelligence and algorithm with, with our next speaker, Dr. Divya S. Dr. Divya ma'am is a senior application scientist in National Support for Bioware Discovery Studio, LTIM Technologies. She was a gold medalist during her post-graduation from Satyabhama University. She has completed her advanced postgraduate diploma in applied bioinformatics, University of Medicine and Dentistry of New Jersey, and SLRIS, now which is known as BioWare. Before Altim Technologies, she had worked as application scientist for life sciences, Apsara Innovations, Bangalore. She also served in DSIR lab and research foundation for the USA collaborative projects in chemoinformatics. She is an expertise in molecular modeling of proteins and transmembrane structure and ligand pharmacophore based drug designing, molecular dynamics, and simulation of lipid systems. She has more than 20 international publications in Scopus indexed journals, including Biomed, Research International, Elsevier, Taylor and Francis, and also two book chapters published by New York Science Publishers, USA, and elsewhere. She is a reviewer of Asian Journal of Pharmaceutical and Clinical Research and also a member of Indian Science Congress. We are delighted to have such an eminent personality to in between us to enlighten our knowledge on artificial intelligence. Over to you, ma'am.
Thank you so much, Aparna, for a brief introduction about me. It's my great pleasure with you all to be on today to see about because artificial intelligence and machine learning are becoming a very popular thing for in a pharmaceutical domains, and people want to screen and how to identify it. So the session is going to be like this. So first, I'm going to show you how to construct your algorithms or in a protocol for your analysis, followed by, after constructing your algorithms, how to execute that algorithm. That I'm going to handle out, and I'm going to show it using a tool called pipeline palette. That is one of the tool under the BioVR Discovery Studio. I'm going to construct the pipelines and going to show you the output. And basically I could say that rather than following other people tools and taking out and doing analysis, this kinds of analysis like introducing a machine learning will definitely benefit to pharmaceutical domain. Let's move on and walk along with me, and feel free to ask me queries at the end of the session. Thank you, everyone. So machine learning and artificial intelligence in drug discovery, because that's why I just given an introduction just about it. What is how every time when we hear about it nowadays, I think in the past 5 years, the machine learning is gaining a very boosting technology in pharmaceutical domain. How to use this? Say for instance, if you have a many, not some many quantities of molecules on your hand, if you want to analyze and to identify which is the best molecule for your analysis, and then how to see that molecule is having really a potent activity or not. So all these things you can able to analyze without going for a different techniques called docking or dynamic simulation. Just by introducing your molecule by the trained algorithms or by constructing a new kind of an algorithm will give you the solution. That's an idea of introducing machine learning and artificial intelligence. Let's work on. About us, Altem Technologies has a company have been for more than a decade as in leaderships in various domains. One of the domain in the life sciences department is a BioVR Discovery Studio, and you can also see the left-hand side the Altem Awards. And this is a BioVR Discovery Studio with scientific portfolio. We have started from a scratch to an end to an various domains, right from an electronic laboratory notebook where you can replace your paperwork into an cloud computing based applications where you can store your data, and then process production operations where we are dealing with various dynamic mapping and then constructions and then bringing out your monitoring dashboard based analysis, and more into a quality and regulatory management where anytime we develop a protocol or a tool, it will undergo certain processes actually. It will test it, and then we'll be seeing how effective it is working out into the things. And finally, we make a documentation and moves on to the further analysis. And in the collaborative science, from our predictive models to when you have a sequence or you have a model, you can go for predictive analysis. And if you're interested in antibody modeling and you want to screen some kind of a macromolecules, of course, you can go into the novel therapeutics. And the people who want to work it on engineering materials like a carbon nanotube or liposome-based drug delivery or a polymer-based drug delivery, we can go for a material studio and more into the chemicals and formulations, screening out the best non-toxic or non-carcinogen or non-mutagen compound for formulating your any kinds of products, maybe in a biofertilizer or maybe a cosmetic products. This is about your rich scientific portfolio. Directly moving on to the artificial intelligence and machine learning importance. So let's see that what is the importance of learning a machine learning and drug discovery. So if people says about it, you have to think about it because we have an intelligence to say this one is not good for us and then this should be a good for us. So we are predicting in such a way good and bad, which is good, which is bad. So we are trying to able to differentiate in between them. So instead of that, doing it manually, we are giving in a program to differentiate which is good and which is bad and to differentiate between the different patterns. So machine learning in the branch of artificial intelligence will really help you, the system, to learn from the data. So you are trying to giving the learning the data and try to identify the patterns. So which should be the good for this particular analysis and how I should take. And based on the patterns, you can make a decision with the minimal human intervention. That is an idea of your machine learning and artificial intelligence. And this one, artificial intelligence, will help you in your drug discovery in many ways. For instance, you want to screen your drug candidates through a combination of machine learning models. There are lots of machine learning models are available, like a random forest, k-means, hierarchical clustering. So different models are available. So which model you want to use, how to use, that is an important thing that you have to optimize for selecting your drug candidates. And this artificial intelligence with an ML method will help you to pre-screen the covariance in your pharmacokinetics and dynamics data. So even I'm going to show you today this also, how it is going to screen and identifying the potential fingerprints of your molecule and comparing to say, if this is a toxic, don't take this molecule for further analysis. And this molecule is having a carcinogenicity, don't take this molecule for this analysis. So all those things we are previously trained and we are going to use this prototype and going to screen with the molecules. That is an importance of using your machine learning and drug discovery. And then this method has been utilized more often in the discovery rather than development. So under probably 1 or 2 years, or I could say that because it's a fast pace of research is going on or carried out in a machine learning. So in future, I could say that there will be a development will happen very soon so that we can make a very fast research and we can identify the compounds soon for a personalized medicine also. Learning. So how we learn this? So we should know about it. We are telling about a machine learning, how we learn for a scientist. There are two different approaches are available. The first method is a pre-built model approach. That means someone has developed this approach. Maybe a person has developed this approach and you are trying to utilize that approach and training with your molecules. Sorry, testing with your molecules. So machine learning specialist who is a data scientist or a person who is working on a very strong in a computer science or an information technological field has developed in a scientific domain to build a models for a public or proprietary data sets or to deploy the model of scientists. So we are trying to use their approaches or a pre-built model, and scientists will apply this model to their problems. So very famous things, if you come across for many people who have no for this drug discovery is in a QSA. So QSCR is a quantitative structured activity relationship or a QSCR workbench, QSPR toxicity relationship or QSPR property relationship. All these workbenches are previously available that has been developed by someone and we are trying to utilize it. And even we have a top 10 model that is nothing but an identifying a compound for a carcinogenicity and mutagenicity. and even for imaging a crystal models. These are the certain examples to speak about pre-built model approaches. This model, you no need to understand much about your machine learning, but you have to know only the processing, how it is getting processed, how to interpret the results. And the second approach is the one people are now trying to do this. You have that means you have to do it by yourself. So we call it a DIY approach. In this, scientists or engineers will try to acquire the data and then build the models from these data sets. And they'll try to apply to their own problems or deploy them to the other scientists. For instance, we are having in a pipeline pilot, we are having components rather than tools and all those things we call as a components. We call, okay, call this component random forest as a one component, and then SVMs, support vector mission, as a one component. We call those components to perform these operations to give by this. But however, you need to require some understanding of a machine learning by a scientist. So these are the two ways you can approach your machine learning for your solving different issues. Okay. How to fit the purpose of approaches? So what is the question? So anytime when you're involving in a machine learning, you should ask yourself, what is the drug discovery and development needed is? What machine learning methods are more appropriate to address this need? And what data can be used to support this machine learning methods? So these three things must be addressed by yourself before getting into any kinds of machine learning or an artificial intelligence based programming. So how do we do this? How do we approach this? So here are the solutions for you all. What is the drug discovery and development? Why artificial intelligence is needed here? Nearly, you will never believe this, an estimated of 2.5 trillion bytes are created each and every day. However, initially there is only one method, or I could say that there are very few methods available to develop compounds and identifying, but it is not like that. Now the quantity of data is keep on increasing, and the people who is doing research is also increasing. The intents of work is also increasing, and the data is created is also intense. So if you have an intents of data, how to interpret and correlate and how identify which is the best, that is a task. So we cannot do it manually. We cannot, if you take in 10 data and do this, of course we can do that. If you have more than crores of data, it is not feasible to human intervention. That's why artificial intelligence are playing a major role in drug discovery. Some of the examples for you all, like a chemical structure data. Now, if you remember, if you have only one database, has been 2 to 3 databases, so people can mention it's PubChem, Zinc, or from an supernatural databases like that. But now there are many databases that keep on emerge, and then they're also having certain could say that for different activity, different compounds are there. So now you can have a more feasibility of accessing to this data because of introducing these kinds of algorithms. And gene expression and genetic data, for many people which gene is expressing and how it is getting expressed and at what level is this expressed, those things they can see by using a microarray in a previous a decade before. Now it has been replaced by a next generation sequencing analysis. Patient specific gene expression you can observe to that, and high throughput in vitro data. When you're screening a lakhs of molecules, how do we identify that which is the best and how it is the best? If we see to the imaging techniques, of course, that is one of the way. And can you able to see to for all the 1 lakh of compounds? It is not feasible by human methods. That's why we are introducing artificial intelligence. Clinical trial data, electronic medical records. It's a very huge healthcare domain where the people are introducing this artificial intelligence, and freely available public data, any data that you're trying to collect from external websites and trying to develop the model, that are also keep on increasing. Commercially available data, internal companies data, data shared among the participating institutions. So all these techniques or certain examples for you to say where the large data, or I could say the big data analysis, BDA, is keep on increasing, where it could be solved only through introducing a machine learning techniques and then the different algorithms to participate to resolve issues. and more intense into a drug discovery and development. So we are how we can introduce this artificial intelligence. You can go for a drug development process and what are the strategies, and then how we use this artificial intelligence. And some of the applications of artificial intelligence drug development, it include past decades applications I'm going to show you, understanding the pathways and finding a drug targets and finding a hit or leads or a synthetic, because synthesizing is becoming a very challenge when you want to screen and you want to take your molecule and after that to see a very good yield of the compound and the mode of action of your drugs, drug repurposing, polypharmacology, and selection of population for clinical trials. These are the techniques or applications where we can introduce your artificial intelligence in drug development. This field is keep on increasing, but it was not static. I could say that there is no static for this. We are just a burgeoning domain. I could say that people are trying to introduce various things and brainstorming and try to identify the various techniques and methods into which. So first, understanding the pathway of finding the molecular drug targets. So first of all, you want to see which drug candidate or you have to identify what is the diseases actually occurring in the patient, how it is get occurring. So for that, we be using a genomics and proteomics approaches. Genomics, for instance, I could say that the RNA sequencing data analysis or an chip sequencing data analysis or even whole genome sequencing. So any data in terms of a sequencing. And proteomics, where we'll be introducing a drug discovery and development like a structure-based drug designing and ligand-based drug designing and identifying which compound or even can identify through the mass spectroscopy which region is getting exposed and you can observe in a difference in between the patterns of different proteins and potential drug target proteins. That is again very important. You cannot target drugs as such, but you have to study the pathways. That is most important, whether it is acting in a downstream or it is acting in a where exactly that messenger is sending to the target to make a disease to be get intense or to be a behavior, abnormal behavior or aberrant behavior that we have to identify. For that, you should know the target. If you know the target only, you can treat the disease so that you can reduce the aberrant behavior of this particular diseases. For that, we cannot use normal chemical pathways. Even your cake, if you open and if you can try to locate and associate pathways, if you put it in that, if your table will be full of pathways. For that techniques, we are using an artificial intelligence and gene disease associated data. You are trying to take some animal models and you're trying to see the disease relevant to the phenotypic changes as a greatest predictive power. So these all are the things where you'll be trying to introduce this artificial intelligence. So you can see in the graph over here. So this graph will says, so what are the number of targets has been taken and what are the predictive, non-target and predictive targeted. The green color highlights a target and then that purple color indicates a non-target. So when you have see the stages of your clinical trial data, in the preclinical testings, it seems to be in a predictive non-target is less. But when you go for further stages, you can see that before pre-registration, the targets of a predictive is increased and then it is decreased and registered. And when you go for launched, you can see there is a number of targets of being keep on increasing along with the non-target. And if you see about in a suspended or discontinued or withdrawn, how many are withdrawn? Because not all the drugs has been approved by FDA has been currently in the market. Some have been withdrawn. So how it has been withdrawn? Because of having a non-targeting interactions with your molecules or in a particular drug targets. So how do we address this? Say for instance, it is a very good paper. If you have a time, you can look into it actually. So this says that we'll try to take an action non-target and an action target and try to generate this as a training set. And similarly, we'll take this as a test set and we apply these algorithms. when you do this kinds of an analysis, finally you can draw the conclusion which all are the targets that you need to analyze, which are the non-target. So specifically, you can pick the targets and you can treat the diseases. This will reduce the time as well as your energy, as well as it will accelerate your discovery process. This is one. The second one, identifying the hit or lead. You can go for enumeration techniques like a molecule enumeration, and you can identify the target specific for virtual molecules. Say, for instance, I have a more number of molecules I have been generated. Using in a ring strain rules, I have been taking only the 15,000 molecules of a starch with hydrocarbons. And if I try to reduce the double bond strain rules, I will get these many molecules. When I apply the valency rules, I am getting 1.7 billion. When I apply for functional groups, I will ended with an 226.4 millions of molecules. So these many molecules will be generated virtually, and we have to identify among them which is the best. So target specific virtual molecules are more important, and this will reduce accuracy rates and R&D expenditure, and it will help you to prioritize the molecules based on ease of synthesis. And in silico virtual pacing technique, like you take your molecules and go for an ADMIT screening, TopCat analysis, and rule out the molecules which is having an behaving an abnormally, maybe a non-carcinogen or carcinogens, or not obeying, not penetrating in the blood-brain barrier. So this method will help you to screen and identify the hits are the molecules before going for your pharmacokinetics and dynamic properties. Lead or the one which is obeys of pharmacokinetics and dynamic properties. This is one application. And if you are a medicinal chemist, I think this is an application for you. Retrosynthesis, that you are trying to identify the word synthetic route and want to success with the roots. So in this technique, what we will try to apply, we will try to use what already the artificial intelligence platforms are there. We'll be introducing three types of deep neural network along with a Monte Carlo search based things for your computer aided organic compound synthesizers. So first take your target and then check for the some kind of add some moieties and do for some reactions and see its output, whether it is succeeding or not. And try to add another moiety in a different location and see for the whether reaction is going specifically to the position or not. So this kinds of a mechanism will help you for predicting your best sort of reactions by minimizing out of scope of compounds. So this is also the one of the application of your artificial intelligence. Predicting. This will be a very interesting tool because when you give your molecule, you should know just maybe lakhs and lakhs of molecules you want to give. It will predict and give you which is absorption level, how much absorption level, good or moderate. And this is a dashboard things which is already present in a pipeline palette. So when you give your molecules, this report will be generally automatically generated. In case if you want to introduce certain changes, yes, of course you're free to do that operations. That's a application of your machine learning addressing in two different ways. So when you give your molecules, it will try to identify how much molecules in your given database is having absorption, good absorption property, or a good aqueous solubility, or it could penetrate to blood-brain barrier or not, or somehow what is going to be what are the molecules are going to have in a plasma protein binding and going to reduce the bioavailability, and what are the molecules are going and inhibiting your metabolism, that is CPY2D6 binding of families, and how many molecules are showing a hepatotoxicity or making a toxic to your liver cells. All these predictions you will see in terms of a visualization, and through that you can able to interpret your results. And then of course more into an genotoxicity and QSA approaches, because TopCat models are always built by using an QSA approaches or QSTR approaches. So this is also the predicting a mode of action of compound using an artificial intelligence. And then drug repurposing. So when you have a new drug in a phase 2 trials to show an indication, so you can take your picture of your, if you have a neuronal diseases. This is an instance, they have some neuronal diseases. You want to predict what is the pharmacological property of your drugs, how it is getting behaved during the reaction of drugs, that also you can able to capture. This technique we call as an imaging technique, where you can identify and you can see the adverse effects of your drugs. And you can also use an algorithm called I could say that they'll be genetically adverse networks will be utilized. And when you design your drugs based on this kinds of analysis, will have a fewer side effects. And you can also try to distinguish between a cardiotoxic and non-cardiotoxic, or a neurotoxic or non-neurotoxic drugs. So it depends upon the gene expression, as well as based on the location or a tissue, you can able to identify the introducing your drug repurposing. There is also other way where you can take an already available drugs that is in the FDA site, you can download, and you can put it your molecules in such a way along with the various transcriptome data, and you can see these interactions using a certain, you can call any algorithms, machine learning algorithms for that, maybe an SEM or an I could say that any supervised learning or non-supervised learning algorithm, and you can predict and you can see which is best, which is having a fewer side effects. So in artificial intelligence, having a very huge applications in drug repurposing. Moving on, polypharmacology. So this is a one I think pharmaceutical people are more interested into it. So one disease, multiple targets. So usually we'll be seeing for one disease, maybe a cancer, there are multiple drug targets are there. Even if you take a estrogen, breast cancer, estrogen receptor, HER2 positive, negative, like that, you can mention the multiple drug targets. How do we address this problem? That is one of the major tasks. And even if you took about it, you try to take a database of molecules using DNA training method, and then testing your molecules for deep neural networks and seeing that which are the drugs are having a good interactions. And you can, if you want to reduce your dimension of your data, always use a PC analysis, that is a principal analysis, will help you to reduce your data. And then you're trying to get a compound. So which is the best, which can be utilized for further analysis. So in order to probe your studies to identify your molecules for multiple drug targets, artificial intelligence are more important. And apart from that, you can also see here, it will help you to probe the data sets of molecules for pharmacological agents, and even you can see the drug-to-drug interaction and associated metabolic mechanisms. Say for instance, maybe A, B, C drugs are there, and then I'm trying to take on multiple drug targets, maybe on A1, A2, and A3. Maybe the drug which should be specific to an A, it is showing its interaction with the B and C. That also you can able to see by using these kinds of techniques. And you can also predict alternative drugs. So instead of using this kinds of drugs, which is going to make a negative health effects, you can bring some other drugs by changing a fragment or replacing a fragment. So this kind of a method or a technique that you have to introduce in a polypharmacology to treat the dreadful diseases like cancers or even Alzheimer's. So what are the things, diseases associated with multiple targets? And this is one of the solution or a way you have to address this. Moving to one directly, these all are the applications, how to introduce and different in a pharmaceutical, I have shown it. So what directly going into one, with how many methods or protocols or components are present in pipeline palette. So right from a basic statistical analysis for a learners, you can go for a categorical big data, genetic function approximation, PCA, principal analysis, random forest models. And if you want to plot a graph by means of similarity or by select a molecules based on distance, or if you are the very strong in R scripting, you are very good in R scripting, then you can use it from an R statistical analysis. But for pipeline pilot, there is not required for any R scripting. You should not have a master's in R scripting, but basic of R scripting knowledge is necessary, because when you want to install the library or to install the dependencies of libraries are more important in your any R scripting. You want to do an GLA model or a linear logistic regression, neural network model or PLS or principal component. So all these things, we are just going to take an input, and then we are going to feed you already trained one, and then your input, and we are going to call one of these algorithms or component to perform and show whether this molecule is having effective or not. So that is, I'm going to show you all shortly. And then if you want to be R-based or native learners, you can have a differentiation. So learners, R-based learners can use this kinds of components, or if you're native learners, if you're very good, then you can call this one. Both are same and behave in the same. That is the idea. But if you're very good in R, you can choose this one. Machine learning. So what are the different methods or machine learnings are there? Supervised, if you take, we have a regression, ensemble, and classification based techniques. And if you are going to take unsupervised, you have to use a clustering, pattern search, and dimensional reduction. And reinforcement, where we're using a clustering and association. So what is the supervised? What is this unsupervised? What is the reinforcement? So I will tell you in a very layman terms here. So before that, what are the methods are more appropriate to address this? There are lots and lots of methods. You can see here under supervised learning, the classification, k-means, support vector missions, decision trees, maybe bases. If you take a regression, logic, linear regression, and then lasso regression, polynomial regressions. If you take ensemble, X boosting, and then ADA boost and cat boost, random forest models, backing, and deep neural networks. So we can see that this is again big neural networks are there. And if you go into unsupervised clustering methods by means of a k-means, so it's a key nearest to neighbors. This is a k-means, sorry. And then hierarchical and a fuzzy algorithms and pattern searches. And you have a dimensional reduction space with a K, T, SN, SNE. So all these different methods of PCA, principal component analysis. So lots and lots of algorithms are available. So when you want to first initially start with ML or artificial intelligence, which method I have to choose and how to choose and how to progress and what is my input and how do you process, that becomes a very huge things will always crack our mind and ask us to answer these questions. Of course, I will tell you these answers for you all. So this method can be maybe involved in fitting of various data and models, and they may be a very heuristic in nature. Unlike a traditional model fitting, you want to fit your molecules like a linear regressions. This is a very good in making accurate prediction for any new data that we are going to give. So let's see what is supervised and unsupervised. When we use this, that is most important and common term that we all should know about it. Supervised learning. This is a model. So in a supervised modeling, when you want to train your model on labeled or given data set, so your data will be split into a test set and then training set. And then we use a training set is used to train our network, and test set will act as a new data for predicting your results. So from that, you can predict the accuracy of your model. So let's say that this is your input data. When you try to update the different algorithm, and your training set is giving this information in a neural way. It is captured already, it has been stored. And now we are progressing it. We are progressing and processing it. We have our different inputs we are trying to give. Once you process, it will try to select the molecule only which is having already given by a training set data. So the robot will identify only the one which is having a more appropriate. This is on a base for any supervised machine learning. Whatever algorithm you're going to choose, that is not a matter, but this is a basic thing of knowing a supervised machine learning. So what are different, some of this one I mentioned here is a linear regression, support vector missions, and random forest. Now, moving on to unsupervised. In the unsupervised, so information of is used to train is neither classified nor not labeled in data set. We're trying to take up molecules from various sets, trying to take it from maybe one is a thioglycine, another one is a metformin base, another one is maybe having a glycoside synthesis, whatever is it. You're having a raw input. So this unsupervised learning will help you to study how the system will infer the function and describe the hidden structure from the unlabeled data. So this is your raw input you are trying to use, and when you try to apply your algorithm and process it, it will do it automatically using which algorithm you're using, that will output your data perfectly. So this is a most and common one that people will try to utilize it. So some of its unsupervised learning of k-means algorithm, principal component analysis algorithm, singular value decomposition algorithm. These all are the technique, but what we need to know in unsupervised, You can give any raw input data of a different systems, and that will be when you apply algorithm and process, you get a perfect data aligned together. That is an unsupervised learning. Now, finally, we have a reinforcement learning. It automatically helps. Say, don't know anything about it, input data. It will reward and give the environment and finally forms and gives you the best one. So it doesn't have to be labeled data set or result to be associated with data. The way it performs and gives your rate, that means you're reinforcing and trying to get your data. So these all are the common terms or common thing when you want to learn or want to expertise yourself into the machine learning. Directly going to the pipeline because we are having a less time to explore with this. The pipeline palette menu, this is a tool looks like this. And we have a menu, search bar, help text and parameter. And this is a protocol view where we are going to construct a protocol, or we are going to call in a components to construct this. And this is a protocol, and we call these all this green blue color are called components. And if you're not sure about it, and you can type which component you want, that will gives you and says you where the components are present. And execution will happen like this. It always moves from the left to right. This is how the progress 1, 2, 3 will goes on. And once it's finished, it will go from top to bottoms. And the first step, so if you want to select your molecules for a learner component, first you take your SD reader, we call as SD reader or SDF reader, call the molecules and learn the molecule using a genetic function approximation and see how the results are there. This is the first step of modeling your pipeline file. So in case this is already built a component with a machine learning algorithm, if you want to edit, you can right click and you can edit, or you can make an alteration in scripts. If you're not sure about it, don't do that. But if you want to do by introducing certain changes, you can do very user-friendly using this kinds of tools. So when I give this, I get results like this. It will predict R squared, R squared adjures, and decimal RMS, and what are the terms are used, and how much coefficient, what is the standard error, and then the p-values. This is a simple model. And then the step 2, use a model component created to learn the predictions. So the model is created in the step 1. So we are trying to call from that component. And then we are asking the molecule to see what is their plasma protein binding for this. So when you ask, the learned component will now behave and then will help you to call your prediction. This is a making prediction with the model in a pipeline palette. This is the second step. The third step, there are 2 ways you can make a model predictions. So first thing, you have been introducing a data and then you are predicting it. If the model output parameter is calculated, you can take up with the function of the pilot script. Here we call as a pilot script. This is a one way of a script. This is not as tough as like another programming. Just a CV, calculate this for log-free properties and then calculate by means of applicability. This is by means of users. You want to call the expression and then asking the tool to calculate. If you're not sure, you can bring the component and you can do this. That's what I told you in the beginning. Either use the one who developed, or you can use a who build your own and they can utilize of your application of your own to your own applications. Either way, we can process this. So now I'm going to choose one by one. I'm going to choose how to design a pipeline. Then I'm going to run, because running it quickly, I think hardly 5 minutes, I can show you all the protocols actually. But we should know why we are using and how to use. That is a very important application of the machine learning. Navy Bayesian classification. Say for instance, you have a molecule. Basically pharmacology people or any chemist people, we deal only with the molecules. So if you have more molecules, and molecule will have a different features and different functions. So I'm trying to take a molecules, feature 1, feature 2, feature 3, feature 4. And I'm trying to see in the same molecule, some features are very good and some features are very bad. That means that could have certain side effects, or it could have an steric interactions, or whatever the other bad in the sample. So now if this is a 1, how do I say which is a good feature, which is a bad feature? I didn't know much about that compound. And how do we say that? So for that, we are going to introduce your machine learning algorithm, Navy Bayesian classification. So you're trying to classify. When you go for classification, use a classification algorithms. Okay. So Navy Bayesian classification. When I use this, try to identify the feature and score my molecule. And from the scoring, I can draw the conclusion. That is one of the best method. So when I try to take a ln of f good, features of good plus 1, and number of features of good and number of features by total, and total to put together, I get the results. So Bayesian models are always binary. It like you want to calculate between a malignant versus in the beginning of tumors, or you want to see the spam versus non-spam emails. So it is not only applicable in a pharmacology, even in your emails, when you try to open your emails without knowing to us, artificial intelligence are playing a major role. Spam mails will be separated, or non-spam mails will be separated. Some will be highlighted, some will not be highlighted. All these things are already trained by that websites, and then we are getting results like that. Same thing even if you want to apply for your compounds also. So you want to see active versus inactive compounds. So what is a good and what is a bad? You can, if you want to differentiate and want to see it in the imbalanced training set, which is having a good in a minority classes, you can learn the good molecules with a predefined molecular descriptors. So just simple one, I'm taking a molecules, the SG reader, and then trying to see the activity class that is already defined. Take only those molecules I'm feeding here and give these molecules and try to identify the which is top good and what are the bad features. So when I give this, you can see the scoring functions that itself is say it's good out of certain molecules. It's a bad out of so many molecules. So which feature of your molecules, which fingerprint of molecules making good and bad, you can able to identify. You can see here, this is a good 13 good out of 47, but this is not showing like a 0. So scoring and seeing the visualization technique will help you to identify the molecular features. So this is one of the way where you can identify this. Second method, say for instance, you're having a molecules, you are working on a cancer. Let me say for instance, you are working for estrogen antagonist, and I have a database of molecules. Someone is giving me a lakhs of molecules. I will never go, they are telling me they should not go for docking, I should not go for pharmacophore, I should not do anything. But you should predict me within a day, or I could say that within an hour, you have to say me which molecules are estrogen antagonist. How do you use that? So we are having a solution for that. We are going to introduce a machine learning, smart way of introducing this algorithm actually, I could say that. First of all, take your molecule as a training set and say these are all the active molecules. And then don't construct this pipeline first. And try to learn and understand the molecule's functions and how it is behaving. Now what we are going to do, you are going to construct the other pipeline saying that your input molecules, maybe the person who has given me the lakhs of molecules to me, and mark those are the estrogen antagonists. So when I'm trying to mark and putting all together, this method LDA, linear discrimination analysis, will try to learn this molecular descriptors and shows me the output, the fit value. So the second plot will help me to show about it. It will call you this, call you the fit values, and then plot the regressions, and then try to identify the A log P values, what are the fingerprints, and then the molecular functions, and then it will draw the conclusions. Out of 500, how many are active? And then you get a ROC, recursive operating characteristics, that shows you the specificity and sensitivity of the molecules. From there, you can come to know which molecules. So this is a first. Second pipeline. After finishing learning this, I'm trying to call that is in a shortcut and introducing a molecules and introducing a false negative, false positive tool. These are the passports, and then calculate the molecular table and try to give. So for this, you don't need to give anything. Just input your source, which is already there, is antagonist test, and then your molecules. That means how many molecules that you have got. And once you run this pipeline, you get the results like this. These all are false negatives. These all are false positives. False negatives. So it says which is active, but it's definitely an R antagonist inactive. But a false positives, it is inactive, but it shows an active. So you can come to draw the conclusion of false negative and false positive. So this is the best way. You can, it is not limited. You can draw, you can screen with any number of molecules. This is the first algorithm, linear distribution analysis. Same component, same analysis. If I want to do it within a deep neural network, DNN method, rather than going for a R neural network model. So what I'll try to do, I will try to consider this model, same input and then construct, but instead of LDA, I'm using a deep neural network and trying to resolve. So what is the advantage of this? It's a more advanced method for building a model and much faster than a neural network model. And it will specify the hidden layer. When you see this, I don't have a time to explain you this algorithm because you have an input and hidden layer and output will be there in a neural network. If you simply type that one, you can get that option, that diagram will be there. And enter the single number and his hidden layers will be processed, and finally you get the output. So just you have to input your molecules, that's it, same one, but we are using in a different algorithms, that's it. So it will try to fit all the fingerprints, and among that it will try to calculate and use your ROC values. So now you learned this neural network, I'm going to apply now. Antagonist. So instead of here, I'm going to call it our neural network model. All the pipelines are same. So initially constructing pipeline is only a task, but after that we want to change algorithm that you can do. And from this, you are trying to change different ones will help you to see which is the most appropriate one or how much a consistency you're trying to get. That also you can calculate and you can draw the conclusion. And you can come to know both the false negatives as well as the false positives. And then so I will show you this as in right now I'll show you all those things as an it will take only a few minutes to run and show you. And applications, it's a very important and known to you should know us. So this is identification diagnosis and personal treatment and behavioral modification, drug discovery and manufacturing. Manufacturing, we consult the drug synthesizers in the clinical trial research. That's the first one I discussed, radiology and radiotherapy, smart electronic health notebooks, and epidemic orbit prediction. So we also developed the SARS-CoV-2 prediction. If you give the X-ray structure, you predict and shows you whether they're having a SARS-CoV-2 or some other pneumonia like that. So especially if you want to see that the imaging analysis will help you to identify and which shows a symmetrical. This is for skin cancer. It will help you to see which is having a normal symmetry, which is a border symmetry, and in one color like that you can able to see for based on that the coloring variations you can give the patient specific treatments and for them to get a cure of diseases. So this is identification. So this is identification diagnosis using an ML method. In 2015, it is reported that 800 medicine vaccines can treat for a cancer bearing trial. So potential applications, you can see that for an cancer, precision medicines, and you can develop a diagnostic for therapeutical medicines. And especially you are seeing your facial expressions, right? This facial expressions will help you for directing a sensitivity to enhance of recognition of negative emotional faces, and then the blunt recognition of positive emotional faces. The depression of patients. See, if you want to see the depression of the patients, just by looking and taking their facial expression data analysis, you can prefer them at drugs. That is also one of the method of implementing the artificial intelligence. And fittest. So today our drugs are cancer patients, example colon cancer. We are giving this therapy. Some are getting affected, some is known effect, some is adverse effect. But tomorrow we are going to depend more on NGS technology, next generation sequencing technology, where you're going to collect the blood, urine from the patient, identify the biomarker, and treatment with the specific drugs that we call as in a personalized medicine. So here we are using a massive amount of your machine learning algorithms. Personalized treatment and behavioral modification, that's what I was saying. The somatics or skin visions, you can use these kinds of technologies to improve and you can treat the peoples. And drug discovery and manufacturing. So machine learning is a preliminary or drug discovery as a potential for the various initial screening of a compounds, and you can predict the success rate based on biological factors and like an extension sequencing analysis. So this is a hidden similar. This is similarly, it looks like a hidden Markov deep neural networks. You have an input nodes and hidden nodes will be there. Finally, form the output. And precision medicine, this is one of the major application of pharmacy people are trying to develop, and they are trying to identify the multi-factor diseases and to turn into an alternative pathway for therapy, and seems to be on a frontier in the space. So I will show you the demonstration. It will not take, it will take very less time actually. So once we know about this techniques only, we can come to know about this technology, how it is adopting. So you can see now, I think you can see my screen. Yeah, I'm sharing this pipeline palette. It's very simple. So it's already built. I'm not going to use anything. First, I'm going to learn this algorithm. So input molecule, it's already there, which I use for the training. And some molecules from NCA drugs I had downloaded. And then I'm going to give it a. You can see how much time it's taken. Yes, finally we got the results. Yes, you can see the results. So it's plotted, and finally you can come to know about how many actives and inactives. And then from this, how many molecules you are given, from that how many inactive and active molecules, you can be able to draw the conclusion. What is the recursive operating characteristics is a 0.96. It seems to be good. It will be able to predict it perfectly. Now going back. Okay, now I'm going back to the pipeline palette again. I'm going to use this model now. So previously I was learned the model. Now I'm going to use the model. Run this. Already a test one which is taken, and we got the results. That's it. So here you get the final results, which are the active molecules, false negative and false positive. So from here you can draw your conclusion. You can take an n number of molecules. From that you get only the top molecules, which are active, which are inactive. So I have shown with the LDA. Now I'll show with you all with other methods also, deep neural networks. Let me close this. I'm going to use a neural network. First, I'm going to learn this input your molecule and then drug likeness molecule. And then I'm using for an estrogen antagonist. Expression is also very simple actually. Estrogen active, take only the active molecules. And then we were run. It's running. Yes, it's finished. Let me share your screen. It's also all right. You can see here, it is predicted within fit values along with your how many data has been taken, and then final model within how many fingerprints has been taken or recorded, and what are the how many actives and inactives. Those results you can able to identify. So I'm going to use a model right now. So here I'm trying to take thousands of drug likeness molecules rather than 500 with already trained set of molecules. Just give it a run. So constructing, it's really needs some time, but since I am just showing you only the output, how it is there, how it is predicting and giving. So false negative active molecules, and then a score of these molecules, and then false positive molecules along with the ROC curves. Same technique you can use for any methods. I will take you and show you for another one, which is very crucial for a chemist. So I'm trying to read a SPL, which is built by us, not by just introducing the random forest model, but already are going to select this activity that's expression by us. So here I'm going to choose the activity greater than equal to class to be active, and then else it should be inactive. I'm giving this simple expression here, if else statement, and going to run the molecules. So it is running. You can see here, first it reads and predicts and get me results. I'll share the screen. So here I will get a results like this. How many molecules are here? How many are active? So it will predict and give me the results like this. So in this tree, you can see here, this is the inactive. And these all are the actives. How many molecules are inactive based on how many which fingerprints, all those things you can able to predict. Just we should know how this algorithm works. Once you know the algorithm works, just follow the pipeline and then get the results. So this is an example, and I show you this one. I think you can see here, tree one, actual molecules, and then predicted molecules, and what is the ROC score. Initially, I have scored only one by one, but here it will predict in all the trees because we are introduced random forest model. So it sees, and from the tree leaves, what are the nodes, how many things are there, it predicts and gives you results. I hope you all can understand somehow. I will try to deliver about its informations. And if you have any queries, feel free to ask me. I'm happy to discuss with you all.
Thank you so much, ma'am. If anybody has any question, they can write in the chat box. Anybody is having any question, they can write in the chat box. Thank you so much, ma'am, for such a nice briefing on hands-on artificial intelligence and algorithm. Thank you so much, ma'am.
Thank you so much, Aparna, and then thank you for this team for inviting me as one of the speaker to show our results to everyone. Thank you all. Thank you.
Thank you.
Thank you, sir.
Now, moving towards our next session on artificial intelligence in drug discovery and development by Dr. Subhash Ajmani sir. Dr. Subhash Ajmani sir is a professional having over 20 years of experience with advanced knowledge in solutioning of variety of real-world data analytics problems across various business verticals, that is drug discovery, pharmaceutical R&D, life sciences, healthcare, software engineering, and pharmacovigilance. He has contributed in filing patents through research resulting in niche product composition, novel algorithms or methodologies. His advanced knowledge has also been recognized by renowned contributors in the field of pharmaceutical R&D, life sciences, data analytics, software engineering R&D, through his research work published in several reputed peer-reviewed scientific journals and presentations in international conferences. He holds the PhD degree specializing in computer-aided drug discovery. He is also an alumni of University of Portsmouth, UK, where he completed his postdoctoral research in development of new methods or applications to support high-throughput experimentation in imperial chemical industries. In academia, he is a visiting faculty in Interdisciplinary School of Scientific Computing, University of Pune, India. He currently works at Genpact as a data science expert, providing consultancy to teams working on various ongoing data science and advanced analytical analytics client projects. These teams help healthcare and life sciences customers in developing analytical solutions for their business problems in multiple areas. So I request Subhash sir to continue his lecture. Over to you, sir.
Hi, Aparna. Thank you so much for your kind introduction.
Thank you, sir.
First of all, I would like to extend my sincere thanks to the organizing committee and also to Dr. Arun Kumar Gupta for inviting me to share my experiences in this particular platform. And very warm welcome to all the delegates and participants. Good afternoon to all of you. I will just share my screen. Let me know when you can see my screen.
Sure, sir. Yes, sir, it is visible.
Yeah. So I'll just start with, there are various components to what we see in the title of my talk. So we are talking about three different things here. I would rather split drug discovery and development into two parts: drug discovery and development, drug development. These are two different facets of the overall process of the pharmaceutical development. And the third component that we are looking for in this particular title is artificial intelligence. So that is something which is aiding or which is facilitating the overall process of drug development and the drug discovery. That's how I see the title of the topic. I'll go to the next slide, please. So I have arranged the talk right now, what I'm going to give in these different aspects. First of all, we see drug discovery and development, how it looks like, and what is the need and motivation. Essentially, what are we talking about? Then we see that artificial intelligence. It will be unfair to actually talk about artificial intelligence in the absence of the overall broad field of data science, wherein it is a piece of, or it actually complements the requirements of data science. So I would rather introduce in this particular talk data science, and then how it complements, or how AI complements to data science. Then we talk about the role of AI in various phases of drug discovery and development. And lastly, what we do is we try to see the limitations and challenges what we are having when we apply those things into various phases. Now, as we all know, when we see drug discovery and development, this is a familiar picture to all of you. We see that we start with, and it remains for years, it remains the same, only the figure gets changed. So we start with a lot compound library of compounds, and we traverse through a path having some of the molecules, leads, hits. We do the preclinical trials, clinical trials. Eventually, if we are successful, and all of us want to be successful, we get some compound which gets approval from FDA. So essentially, when we see this, and this is how it looks like, I've taken a couple of pictures, maybe these pictures which are coming from different literature reports at various point in time. What it shows is there is a difference in the numbers, but overall things remain the same. And this showcases that there is a specific need to reduce the cost, the time, and the risk involved in this particular process. And this is the need, and the motivation behind all these things is to finally cater to the needs of the patient, making the standard of care and quality up to the mark for the patients. Not only for the patients, it is going to be helpful when we do something streamlining the process. It also will help pharmaceutical industries to have more viable commercialization of the product, and also for the healthcare system as an overall, it is going to help to make the process efficient. Now, we understand that there is a need, there is a motivation, and as a result of that, as you must be knowing, the regulatory authorities are now conducting workshop, going for educating people, and this particular practice of model-informed drug discovery and development, which is in short known as MID3, is being promoted, and it's been looked upon by pharmaceutical industries, by academics, by regulatory agencies in different ways. So if we have to see that, MID3 can be defined as, I'm just giving a definition of that, a quantitative framework for prediction and extrapolation of the knowledge and the inferences generated from various models that we generate throughout the different stages of the cycle, using compounds, mechanism, disease labels, and other things, and which ultimately leads to improving the effectiveness of the decision making, go, no go, at various stages of the drug discovery and development. So this is how it is defined as, and it includes various components of pharmacokinetics, pharmacodynamics, quantitative system pharmacology, machine learning, artificial intelligence. There are various components added to it. And it has been shown to play a very important role in effective delivery of new therapies, and finally leading to decision making internally for the company who is doing the drug development, and also for the regulatory agencies in assessment of whatever has been given by the companies to the regulatory authorities to assess and judge or evaluate a particular product or a drug, prospective drug, so that it reduces the number of cycles, the to and fro that happens between the company and the FDA before it goes through the approval. So this is how this particular thing gives us a background or a platform to take it further and talk about it in terms of the quantitative framework. And that's where there is a role of artificial intelligence and machine learning also coming into play, along with the other domain specific areas, which is more into in terms of the activity, the pharmacology, the pharmacokinetics, and other things. Now, when we see this, we are all familiar with this, but just to reiterate, these are the different phases of the drug discovery and development. Starts with the pre-discovery phase, then we go to the drug discovery, we explore molecules. Once we shortlist few molecules, we go for clinical testing, we go for clinical trials, then we go for regulatory requirements, uh, and finally we get the compound or the prospective drug into the market. So all of this giving that background, what is drug discovery development, what is the importance of expediting this particular process, I would just go directly into introduction of data science and artificial intelligence. So as we see here, I've highlighted that data science is essentially an interdisciplinary field which encompasses many fields, starting from domain to computer science to big data to decision support systems, and artificial intelligence being a part of it. There is a hierarchy of needs for data science. Is essentially, it starts from the data. We see what data is there, then we go to the data organization, data storage, data manipulations. We go for exploring the data, transforming the data. As we go up the ladder, you see that the AI or machine learning and other things comes over here at the top, which is like you go higher in the ladder, you require those things. Data science, essentially, one of the component that is highlighted over here is the deep analytics. That's where you need the statistical analysis, you need artificial intelligence, machine learning, and other things. But apart from that, data science as a discipline encounters or consist of data management, advanced visualization, data protection. There are several elements added to the data science. And one of it is essentially the analytics, wherein we may or may not require artificial intelligence, depending on the problem that we are having in hand. But that's where the AI comes into picture. Now first important thing is, what is data? So I would like to take this basic thing, what is data? So normally what we think is something as a number is a data, but data can be of different forms. It can be number, it can be quantity, it can be information, it can be a graph. All the things that we see is some form of the data. And data can be collected for any topic or anything of your interest. And it could be relevant. Right now we are at a scientific forum. We may be using data for scientific research, but if we go to business, it is also equally important for finance. It is also equally important for business management and other departments of the allied businesses. We all know what is science. Science is the knowledge, and knowledge that we attain through the practices and studies. So when we combine these two things, essentially we are talking about a data science as a discipline, wherein we start from a data. Now, data can also be of different types. It can be a structured data. Normally, what you see or what you collect in the lab or what you do in your regular practices, you mostly talk about as a day in, day out structured data. But most often than not now, in these days, what is more important is, or what gives us more information also is unstructured data, which are collected from the different social media stages. We have got different audios, videos. There are various mediums which also give us the data, which typically feeds in. And then we go through this cycle. So we have the structured data or unstructured data. We convert that into usable format. Then we apply some scientific applications or algorithms or methodology on top of the data to generate the knowledge, to extract the knowledge. We have something in, when we start looking at the data, we have something in the mind. And that's where we need to connect it. We need to get the knowledge extraction of that. Now, the knowledge extraction, most of the people say that knowledge extraction is fine. You end at that stage. But that is important, but that is not it. You have to go beyond it. And beyond it means you have to go and you need to get the things in a meaningful manner. And once you understand what knowledge you have extracted, the meaning of that, you generate it into actionable insights. That okay, this means this, and now I am supposed to do this. So that's where the overall cycle of the data science and overall cycle of problem handling ends. So this is the last piece is very important. Unless you have this last piece, meaningful and actionable insight, you are just at the verge of or at the edge of doing something, but you will not be able to do it. And that's where you need the support. Now there are various techniques to this discipline of data science. It starts from data mining, contextual analysis, probability, statistics. There are various techniques that we use for doing data science problem for dealing with data science problems. There are various application areas, and here when we are talking about, we are specifically talking about the healthcare area, and in that healthcare area, we are talking specifically about drug discovery and development. But apart from that, drug data science as a discipline is being applied into various other sectors. This is just to name few. Now, this is a process. Essentially, you start from, basically, you need to ask the questions. I will say that you need to ask the questions. Data is one part of it, but you need to start with asking the questions what you need the data for first, before you start getting into the data collection. So you frame your problem first. What do you want to do? Okay, I want to generate a quantitative structure activity model. But before that, what is the problem in hand? Don't we have the other models? Is the area what you are trying to make the model for, has it been explored? What are the limitations of the existing models that are there? So you need to actually see and interrogate the problem that is there in your hand. You need to understand that, and then you fix on a particular spot that okay, these are the things that I want to deal with. And once you finalize that, and that's where the domain expertise comes into picture. That is the most critical step. Then you go into the, okay, now I've decided I wanted to reduce a particular side effect because the other models doesn't address that. I wanted to do that along with retaining the activity. Then you start collecting the data for that specific problem. You search the literature, you go for various public proprietary databases and other things, you get the data. Once you get the data, you start processing the data. Processing the data, you just start can't directly go and start building models. You need to see what data do you have, how many of the data are missing values, whether the data that you have collected from various sources, are they in the same unit or not. These are the basic things that you would be looking at before you get into the exploratory phase of the data. You need to correct that, you need to organize the data. Then you get into exploratory phase wherein you see that okay, these are the different things that are available, these many molecules are there. This is typically the collection that you have in terms of the chemical diversity, if you have to talk about. So I'm just taking an example. So this is how you go, and then you start building up your model. Then once you get the model, you need to understand that the interpretation is the another very important thing. If you are in the same domain and you have to explain the person to the same domain, you are lucky. But if the person, you are working for somebody as a data scientist and you are working in a different domain, you need to explain it in a way or communicate the results in a way that the other person can appreciate that. If you go more technical, the person won't understand that. So that is also very important, the interpretation and communication of the results in the way that it is appreciated by the stakeholders or the person for whom you are making it. So this is very critical aspect of the overall cycle. So these are different stages that you undergo for in the data science process. And I would rather not go into this details of this particular skills required, but there are various skill sets, as we have already seen that it's an interdisciplinary field. So at various stages, you need to have various skill sets required. So when you want to do the programming, you need to have the programming background. You need to understand that scientific computing. You need to, when you have to frame the problem, you need to understand the domain. Unless you understand the domain, you wouldn't be able to frame the problem. So like that. So this is just an idea to give you what all you need to do before you get into this data science or application of artificial intelligence. Now, it is equally important to know what all things are needed to do this particular cycle or the process. So typically, as the previous speaker also said, that R is one of the most important or one of the majorly undertaken scripting language. But apart from R, you have got Python also, which is equally important and gives you a flexibility. And these are all scientific computing languages. But apart from that, when it comes to visualization of your results, you may want to take help of some softwares or something which is specifically for visualization, like Tableau, Click, MicroStrategy. There are various software which are meant for visualizations. So this slide just talks about that. Now there are various stages. You may have the problem, but then there are various ways of doing analytics. What do you want to, once you have zeroed down on particular data, how do you want, or what information you want, or how do you want to, what is your purpose? So there are various difficulty levels and the value that you get out of the data as you go in a particular way. So descriptive analytics, diagnostic analytics, predictive analytics, and prescriptive analytics addresses the question, the corresponding question which is given over here in the chart, left-hand side chart. What happened? Why did it happen? What will happen? And prescriptive analytics is the most important, and the most difficult one is to propose based on whatever you have studied that, okay, next move would be this, or something to aid to your decision criteria, or to for taking the decision what to do now. So that is the topmost level thing that you are trying to see with respect to when you start from the zero level of the data. And in doing so, what you do is typically you make use of in the preliminary stages of the descriptive and the diagnostic analytics, you make use of typically statistical techniques to visualize the data, to see what patterns are there. But as you grow on in going from the descriptive to predictive or prescriptive, you make more use of machine learning and artificial intelligence. Now I keep taking the name machine learning, artificial intelligence, and I keep saying that it's a complement to data science. So let us see what is artificial intelligence. So artificial intelligence is any technique which enables computer to mimic human brain. And there are various components to artificial intelligence. And what is typically talked about in terms of the drug discovery and development is more often than not machine learning, which is highlighted over here. But apart from that, you have got artificial intelligence in terms of natural language processing, expert systems, speech recognition, computer vision, robotics, planning. There are various components to artificial intelligence. And in some way or not, some way or the other, they are also being used in drug discovery and overall this particular aspect of the product development cycle of the pharmaceutical also. Like in the previous speaker was talking something about the facial expressions and other things. So essentially what we are talking about is the AI, which is specifically related to computer vision. And here we are talking about image recognition and machine vision and taking some decisions and giving some sort of a conclusion or some recommendations based on that. So, but mostly when we talk about is everybody talks about it in terms of machine learning. Now, there are different kinds of machine learning. I think previous speaker also touched upon that. But just to recap again, there is supervised learning, there is unsupervised learning, there is semi-supervised learning, there is a reinforced learning, there is evolutionary learning, there is a deep learning. These are just to name few, and there are different kinds of machine learning. And machine learning as a tool is nothing but a data analysis method that automates analytical model building. Essentially, going through different iterations, it tries to learn and see what all hidden patterns are there so that it can make something useful and something which gives you some insights in the data. The hidden insights it will unreveal. So that is the function of machine learning, and it's a branch. It's again a node from the AI that is coming out. Now typically what we talk about is the supervised and unsupervised learning, and here also I just try to say that, okay, unsupervised learning, you've got a very layman's term. You've got a set of animals. You say that in unsupervised learning, you've got a set of animals, and you say that you apply a particular method, it will automatically using some features Like the feature could be what kind of animal. So like bird, hare, a crocodile, it separates it out. So there is no thing attached to a particular object in this particular figure, but still it will be able to categorize those things into the similar classes. Whereas the same problem if we take it as a supervised learning, we actually label it. We label each and every object or the animal in this to a particular label or the response, and then we do the supervised learning using some features, and then it gives us a model. When we apply that particular model for a new object, it will say that, okay, which particular class or which particular object type it belongs to. And this is again just to showcase what all different kinds of machine learning and which is relevant to the field of pharmacy. These are all the things that are available and has been spoken about by the previous speaker. So I won't go into the details of that. Now let's go with this background of the problem in hand and data science. How do we deal with the drug discovery and development? So the first step or the first phase in the drug discovery is essentially pre-discovery. Herein, what we do is we normally talk about what is known about the disease condition. And typically, before taking up any project, we need to understand what sort of things are there in hand, whether it's to what extent we understand the mechanism of the particular disease, what is the data available in terms of the different omics that you are there, and most importantly, in terms of pharmaceutical industry to take up anything, they will see that whether the unmet medical need is there or it's a rare disease. So these are the different facets or aspects of the disease that typically you would consider in the pre-discovery phase. Now, in order to see and try to take help from the data and the data science and also from machine learning and artificial intelligence, it's a typical cycle that makes use of this particular data in a way that you go to the public domain or you go to the proprietary databases, you get the data, you start doing some sort of a integration of the different data from the different databases, you do the modeling of the data, you generate some annotations, and this particular thing goes on and on. It's an iterative process. You start from somewhere and then you just reiterate. So this is a typical cycle that we follow with the omics data, and there are various types of omics data that you need to integrate, then you need to process it, and finally you get something valuable which you can make use of in terms of taking a first step. Where and what is the next? What should I take and go where? Which particular disease area, or is this particular disease area which I want to consider, or this particular topic that I want to consider, how much relevant it is? And here is the importance of machine learning and deep learning when you try to churn out the data and try to get the various aspects of it. Excuse me. Now, typically here, text mining approaches are the first one that is used because there is a plethora of scientific literature out there that is available. So you need to make a guess that what all literature is saying. So in text mining or text analytics, using the NLP, NLU, NLG, basically natural language processing, natural language understanding, natural language generation, all these things collectively, you need to make something as a conclusive thing coming out of it, that okay, which ranks you the different topics of your interest and can guide you that okay, these are the potential areas in which there is a likelihood that you should go ahead, and if you take the step further, you will land somewhere which is more relevant to the requirements in hand. So this is typically the first aspect wherein you scan the biological landscape. Second thing is you need to understand the different, there are systematic studies done for understanding the mechanism of the different diseases, but can we make use of text mining and analytics to propose the potential mechanisms? So that's where also to give some insights on the potential mechanism and mechanistic aspects, text mining actually plays an important role, wherein you actually have the different sets of you explore the data set, and using the different articles and the mesh terms involved in that, you do the joint conjoint analysis based on the keywords and the phrases and other things, and then you come into a particular conclusion based on those analysis to say that okay, there is a possible linkage of this particular receptor or target or a pathway to this particular disease based on some analysis that you do available on the scientific literature. And one good tool or one good methodology given is Melody. If you want, you can just explore this. This is very interesting. Another aspect is wherein the social media analytics is very, very important. And Dr. Matthias Michel, who is the text and data analytics commercial lead for life sciences solutions at Elsevier, he just wrote this particular statement. Although pharma perceives some risk in using social media, it's actually riskier not to use it. So if you don't consider this, you are probably not doing justice to the available information, ignoring that particular information. And this typically helps you to identify the unmet need and also the rare diseases, identification of the rare diseases, and particularly knowing the sentiment of physicians, the healthcare professionals for a given disease, and also about the patients. How do they see the available treatments, the available standard of care? So when we go through the social media data, you will come to know about this a lot. And ultimately, using this sentiment analysis and the opinion and views of patients, doctors, and other allied paramedicals and healthcare professionals, you will certainly be able to get something, some direction, some clues that, okay, this is where, again, it sort of substantiate my thinking that whether I should go in a particular direction or not. Typically, typically looking at initiating a particular topic or R&D project. So this is where all these things, when we do, collectively, it will help us in knowing about the disease condition, whether it's a unmet medical need, it's a rare disease, what sort of mechanistic understanding is there, can we propose something new based on the available data, and what all omics data says about a particular disease. What is the favorable, to what extent it is favorable to take it further. So these are few of the outlines that I could say in terms of pre-discovery phase, wherein all the AI and machine learning or text analytics or the other component of NLP, NLG, and NLU are used in the pre-discovery phase. Now let's get into the discovery phase. Typically, when we go from pre-discovery to discovery, what we are dealing with is more in terms of molecules or the compounds. So which molecules will result me in the promising treatment is what is the question that we are asking, and we are trying to see that and try to address that by doing different steps and tasks. Starting from screening, designing, optimizing, prioritizing a set of molecules that you are already having in hand. Now this, I think most of you are familiar with all these things that we do in the drug discovery phase, because in the lab you must be doing all these in your postgraduate and PhD programs. We are taking up, and I also did the same while I was doing in my PhD program. So, but nevertheless, it's better to reiterate that and recap what is there and what are the advancements. So these are the typical data types or the data that we are dealing with in the drug discovery phase. You're dealing with the curated databases, systematically curated databases by different vendors. You are looking at literature and patent databases. You're looking at physicochemical and ADME toxicity data. You're looking at the structure searching systems like SciFinder and other things. You are looking at the various activity in vivo and in vitro biological activity databases like your binding DB, your what is that? Therapeutic index, TBDD. Then, so these are the different databases and data types that we typically use while we are doing the drug discovery stage. And there are various databases which has the data into various forms, And then typically you need to take all these things into consideration once you want to take a collective decision for moving ahead in terms of shortlisting your molecules or coming to something which is like more in terms of uh, potential candidates. So all these things you need to consider. And then you not only typically talk about the compounds, you also need to deal with the biological databases, and then you club them both and then generate. This is all done. This is all done by a great visualization. Visualization helps a lot in linking these two kinds of databases. That's what I will say at this particular point in time. Then comes using, when you have got the set of molecules, you typically, or the set of targets, you typically go for all these methods, probabilistic statistical methods, pattern recognition, and you do either on the biological landscape side and the mapping of it in terms of the compounds or the molecule library side, and then you make a decision, collective decision, what is better for you to take into the next stage. So typically on the biological side, you go for target identification, validation, pathway analysis, pathway enrichment. There's so many things that you do on the biological side. And then you start with virtual target screening, then ligand-based design, whatever is the need at particular what stage you are there. Based on that, you will typically take up the step, you club them, and then you go for the next step. A plethora of methods are available. Whatever gives you the best result is the one that is best for you. There is no one single method that can be suggested based on the best of my knowledge and my experience over the past few years. So there are typically different methods available. You need to apply those depending on what data you have in hand, what problem you have in hand to solve, and typically what disease areas, typically what sort of problem you are dealing with, and then you take it up and then you apply it to get the solution. This is just a snapshot taken from a particular literature which is available online, and which was quite a recent one in 2019. Then we go, after doing all this, we go into the preclinical testing. And here, what we are interested in is typically whatever molecules we have in hand now, can we have something to prioritize those for the next stage in clinical trials? And typically here, what we talk about is the pharmacokinetics and pharmacodynamics, what the body does to the drug and what the drug does to the body. And also, not only this, we also have to be seriously thinking about the toxic aspects over here. And then we go to the selection of the candidates for the next stage. Now, very important point over here to emphasize is we don't want to sacrifice. There is a lot of interest in preventing the life of the animals. So we are looking for alternatives to the animal experiment, and that's where here the data science or artificial intelligence or computer models comes into picture. It gives us in hand some alternatives which can be used for doing some preclinical testing. And with the help of artificial intelligence algorithms and big data approaches, we are able to simulate the activity of many drugs on many tissues, like in a virtual human. So there are virtual organs that you can develop, and based on the virtual organs, you can test your compounds in the virtual organs to see what kind of effects it would probably have on the different organs to take your decision. So here we are talking mostly about the mathematical model and simulations. So either you have a mathematical model or a computer model to assist you with a particular treatment model or a disease model. You take that in hand, you pass your molecule through that, and then you get the corresponding result. Or you do the simulations, and then you say that, okay, typically using the simulations, this is how a particular compound to mimic in the body of the human, virtual human. So that's where there's an important application area of machine learning models or the mathematical models into the preclinical testing. And one of the very precise and concise way to look at it is a framework given by a group of scientists 5 years or maybe 4 years back. This particular framework shows that you have got different chemicals, you have got different targets, and you collectively look at them together. You generate the profiles for each and every candidate to take the collective to get to know the profilome of a particular compound of your interest and see. And this particular experimental validation shows that how close they are able to get it into a particular trend. So that's how it typically gives you an idea about the bioactivity profiles or profilomes for the given candidates that you have shortlisted. So this is the framework. Very importantly, in preclinical testing, predictive biomarker is one of the most important thing that aids in identifying the patient population. So unless you know what are the things that are responsible for a particular disease, you won't be able to take through your treatment specifically to address to that particular disease conditions. So here also, when you have the molecular profiling, you've got a disease category, you just do some sort of a machine learning approach to see that what all genotypings or phenotypings or whatever you have. Typically here what we are talking about is the genotypes or proteomics, what all things are important for a given disease condition, and what is the sensitivity of those different protein elements or gene elements across a different condition, a disease condition. You come to know about the predictive biomarker that you can make use of it later down in the clinical trials. Now, the most important part and most hefty part is the clinical trials. Till this part, you've done a lot of things, and you want to make sure that whatever you have to test on the first in human experiments, you ought to be very good. So we have tried your best till now. Now here, this is a very hefty part, clinical trials, and it has got various arms to it. So when we come to this particular stage, we ask the question, did we find the prospective drug candidate? And with respect to all these things that you see here, they are all relevant aspects we need to see and we need to find out and we need to check. Where do we stand? So in terms of safety tolerance, efficacy, adverse events, regulatory requirements, manufacturing, the marketing and commercialization, that is the foremost and important thing for any pharmaceutical company, because the commercialization is the only thing wherein they will get the return on investment. So when you see typically clinical trials, the most important or the roadblockers or the bottleneck is the patient recruitment and retention. Essentially are the key barriers for the success of the clinical trial. And it has been found that 85% of the clinical trial fails to retain enough patients. 80% of clinical trial fails to finish on time. So the more you delay the clinical trials or it gets delayed, the more financial implications and more company has to suffer for that matter. 50% of the site enroll one or no patient for their studies. These are typically statistics that is available into different reports, market research reports, that shows that there is a lot of things that needs to be done to have an efficient clinical trial stage. And that's where, before we get into that, we need to understand what all things are done into clinical trials and where we can pitch in them. So if we see what happens to data in the clinical trials, because that would be there in our hand, is typically you generate the data of a patient, you collect the particular data from the site, you encode the data, because this is a sensitive data. So data coding has to be done. It has to be anonymized. It may be there in EMR, EHRs, the electronic medical records, the patient registries. There are various ways in which the data is recorded. The data is collated typically into, nowadays it's mostly the thing goes into cloud, but there are, because of some governance and security issue, it's typically also kept into as a backup. And then that is available for us for analysis and report. This is how typically a clinical data is there, and what happens to once we start the clinical trial process. And these are the different applied patient data types, which I was just mentioning about: the EMR, HR records, patient registries, claims data. Because normally in the US, you have got a system wherein you have to go through the payer, somebody, the insurer. The insurer will pay for your treatment along with your site. So you have to pay a part of it. So that's how the claims data also has the information about most of the important things, like the patient history, the disease, the doctor, what sort of treatment has been given, how much he or she is adherent to the medication. A lot many things you would be available into these different patient data formats. When we have the data, what do we do with it? That's the question. So you can do a lot of things with the data that you have in hand. And typically, these are the different sources that is available to us. You can apply advanced analytics, you can apply computer vision, you can apply artificial intelligence, you can apply NLP, you can apply visualization, different things you can apply to achieve to various aspects of it. Especially in terms of clinical trials, if you have to see, you can go for cohort querying, you can go for patient stratification, you can go for pharmacogenomic studies. So these are the typical aspects wherein you are typically looking at the patient population, and what are the population which you need to address, or you need to typically start your application of the drug in the clinical trials based on your biomarker understanding, and based on your understanding of the disease process and these data available to you for the patients. You can have the queries based on your requirement, and you can shortlist the patients, do the patient stratification based on your different classes, and go for personalized medicine also. This particular picture, this is a very interesting picture, and this shows that it starts from essentially from the top. If you see, these are the various facets that are important, that we spoke about earlier. The cohort composition, because ultimately your results will depend on what cohorts you are applying your prospective you're testing your prospective candidates on. Patient recruitment is another thing. Even though you are able to find out whether you are able to recruit the patients or not, that is another aspect, very important. And even after you recruit the patients, whether you are able to retain those patients, and that is also, patient monitoring is very, very important. And it has got three particular aspects: adherence, endpoint detection, and retention. These are the three things, and various machine learning and artificial intelligence methods based on human augmented intelligence also is applied over here with some reasonings. So that's where the role of AI comes into all the different stages of the clinical trial. And these are various outputs that actually it caters to. It maximizes the efficiency of the overall clinical trial process in typically talking about elevation of the problems that we have seen in terms of patient recruitment and monitoring. So these are the things that, and apart from that, it's not only data, it's also the wearables or the IoT, Internet of Things. Along with that, it is able to see the adherence, endpoint detection, and retention, because these are the things which gives you real-time data for how the patients are doing. So that's where IoT, wearables, and the data analytic techniques all together gives us a platform to expedite or to make the process efficient. So typically using the AI, the clinical trials is transformed into more intelligent process. And these are the various aspects of it that gives us the benefit in different terms. So you can do the site performance prediction. Based on that, you can say that, okay, I want to do enrollment over here. I don't want to do, based on the dropout rates of the patients over there. You can have a centralized trial monitoring based on the system that you set up. You can monitor the drug adherence of the different patients. You can digitally analyze. You can get the digital biomarker and the progression of the disease when you are doing the clinical trial. It's like hand in hand. It is happening. And most importantly, it also gives you the clinical data cleaning, which you can make use of it later on when you want to go to the regulatory authorities to showcase the results. So this is all coming into a very nice manner, all collective, so that you can take it to the regulatory authorities, and it will be easy for them also to assess and to judge your case. So that makes them also comfortable, because there are standard formats there. So that's where the whole analytics and the real world data, there are various terms involved over here in this particular page, but they are all important and they have got the respective importance into the overall enhancement of the clinical trials and transforming it into a more intelligent process. Then getting into the regulatory affairs, typically regulatory affairs, if we see, it's essentially around the drug marketing, the drug approval, and across all the different stages of drug development. Because once you have got into that stage, you need to have all the documents in a way that you can produce it in a given format and in a given fashion to the regulatory authorities, because different regulatory authorities have different formats. Although there is a harmonization committee that actually gives you a um, standard format, but still there are variations from different agencies. So you need to make sure that you have all the information that is needed for applying, for filing for your drug as a prospective treatment into different for different authorities. So getting into that one-on-one. So regulatory requirement, you need, there's a lot of text available because it's all related to law, regulation, guidance. So text analytics and mining typically helps you over here to monitor regulatory context and requirement, because if it keeps on changing, there's a monetary, you need to do the regular monitoring, and the system can help you in doing this automatic monitoring based on the different indicators or the key performance indicators or some alerts for that matter. If there is any change, you will get to know, and that reduces, because if it is not there, you need to do it manually. So it reduces a lot of manual effort, and manual is a very Herculean task, going through all the sort of text material that is available from the regulatory bodies. And it also helps you to make decision making based on the earlier decisions that has been made by regulatory bodies for the similar or like cases. You can actually delve all the text or the information available from the previous cases similar to yours, for clinical trial information and for competitive label. So you can take help from all these things from that particular machinery, the AI machinery that you will develop, and it will help you in making a very informed decision based on the available things in hand. And not only that, it actually makes it more efficient and faster and better understanding. So these are the advantages. Responding, so it is not only that you submit the case or the dossier to the regulatory body, it's gone in one go. There are different iterations that happens. There's a to and fro. There are questions given by the regulatory bodies. You need to answer to that. So you can also take help from the existing questionnaires. You can see that what is the similar questions that has been asked earlier. You can make use of that, and typically doing some sort of a text analytics and making the system more towards summarization, the text summarization. You can make use of all the machinery to help you in answering the response, answering to the RTQs from the FDS or the regulatory bodies. There's a lot of importance of doing the large scale manufacturing and how to make it efficient using the AI methods or the data science typically. And there are various indicators that they call that can be used to improve the processes and make the process efficient. I have just lined a few of them. The quality control, the shortening of the design time, reduce material waste. These are, so here typically we are talking about IoT, robotics, and the machine learning or AI system in combination, which can make this particular process efficient. The next important thing is the marketing and commercialization of the new drugs. So as we see nowadays, at least 25% of the marketing is done today is delivered over the digital platform. So you have, when you say something digital, the data is available to you and you can make use of it. Real-time monitoring, we have already discussed. The same thing is applicable. You can monitor the patient's health, and based on that, you can actually make use of those real-time results to enhance the sales of your drug. If you come to know that your drug is doing really good, you can showcase the results. There are various dashboards. There are various KPIs you can give it in terms of very good visualization and graphics, and to promote your sales of your drug. And that will only be possible when you do use these platforms which are integrated on the real-time data manipulation and intelligent data analytics platform, so that it gives you alert and it gives you automatically the visualizations that you can make use of it in terms of promoting your drug. In the same way, you can also use it for the business development and licensing side, using the different models that you have in hand. And last but not the least, the commercial side, for both the sales and marketing efforts, the different methods. So when I'm talking about, let me clarify here, when I'm talking about AI, it may be AI, it may be, because you will not apply AI unless and until it is needed. If the statistical methods or simple visualization tools helps you, there is no need for you to get into the AI or sophisticated machine learning techniques. So always be simple. If the simple things works for you, it's better that you use the simple thing and then go ahead, rather than making it more complicated. But if the things are not getting solved by the simple methods or tools, you need to definitely make use of the sophisticated tool, which may help you in sorting out that particular problem. Post-marketing surveillance, even after doing the drug launch, we need to continuously monitor that. And we are now looking at the vaccine, the COVID vaccine. So basically, it's all pharmacovigilance. That is very important. We are all reading through the reports every day, how it is getting done. So it is essentially integrating the real-time data, separating the signal to noise ratio. So essentially, the pharmacovigilance is nothing but the science and activities concerned with detection, assessment, understanding, and prevention of the adverse reaction to the medicine. That's what is the discipline of pharmacovigilance is all about. So traditional methods, you used to do it by case, by the frequency and the reports rates and other things. But with the advent of computers and the advanced techniques, you make use of it. And typically, it helps you to make it more efficient and more precise, because it helps you to remove more efficiently the noise from the signal, so that you detect the proper signal and then you take the proper measure for that. There's another, there are conferences that are happening for looking into different data mining techniques and how do we support the pharmacovigilance. So this is just a snapshot to show, and there are various data mining algorithms that are supporting health professionals to detect the potential safety signals. So typically, this is just to show that there is a lot of things going on in this particular field, and there is a lot many things to be done. We are just doing whatever we know the best, and there is a lot of more scope to improve these methods and techniques to address to the problems that are relevant in the field of pharmacovigilance, as well as in the other stages. Ancillary areas, apart from the drug discovery and development, ancillary areas would come in drug repurposing. You wanted to do product lifecycle management. A company invested a lot of money. If it comes to the end of there's a patent expiration, they really would like to have it prolonged for further time. So that is the way it helps. And drug repurposing in itself is a very broad subject. And a previous speaker also spoke about that. How can you see drug repurposing or polypharmacology? So various techniques are available which can be used for drug repurposing. You can also do increased, you can increase your networking and collaboration using the different platforms that are available. You can go for synergistic drug combinations for treatment of new indications. Drug repurposing typically talks about one compound or one molecule. Synergistic drug combination can also be an effective treatment and can give you new indications. Drug-drug interactions, diagnosis errors. So essentially, decreasing in the errors of diagnosis, there is a lot of application of computer vision, image processing nowadays, which is aiding a lot into diagnosis. And the one that we are talking about in the previous speaker also mentioning was the snapshots, the MRIs, the CAT scans and all. Typically, those images are being used to diagnose the disease condition properly in different stages. So you decrease the errors in diagnosis. So with this, I'll come to the summary of the thing that we have already seen, and it has been given very nicely by a paper, how AI is involved into different stages of the DDD. So I'll just take a pause here. You can just go through it.
Thank you so much, sir, for such a value addition lecture on artificial intelligence in drug discovery and development.
Yeah. If you allow me 2 minutes, I'll just want to cover the limitations and challenges.
Okay, sir.
So all these things that we are doing is essentially we are having limitations and challenges in this, and we should be aware of that. So first and foremost important thing is the data acquisition. Although we all say that a lot of data is available, but typically, even if it is available, to acquire it and to have it handy for your use is one of the very challenging task. To get it in your hand. And once you get in your hand, you need to somehow, whenever you get that particular data into your hand, you need to somehow deal with the dirtiness of the data that you have. You need to separate it out, what you want and what you don't want. You need to typically work on that. And if you don't know about the quality of the data, if it is, then you are bound to get unexpected results, and that leads to the higher error susceptibility. So you should be very clear in what you are getting, and the quality of the data is very important. You cannot use anything and everything, because in the computer science they say garbage in, garbage out. If you put the garbage, you will get the garbage only outside. But if you know that there's a high quality data and standardized data, you will definitely have something which is meaningful for you. And problem of data privacy, like whether you want to share it, you don't want to share it, that also you need to deal with. It's again a limitation. Sometimes you don't get what you want, but you get what other wants to give you. Apart from that, you may work in your domain, fantastic. If you understand all these data science principles and technologies and techniques, it's fine. But if you are other way around, if you are working as a data scientist, you need to understand the domain. If you are unable to understand the domain, you won't be able to cater to the needs of the problem. The interpretation of the results becomes very much critical there. Because if you don't understand the domain, you won't be able to interpret the results. It will be just numerical for you. Numerical, analytical, but not beyond that. But in terms of business, in terms of the R&D, in terms of whatever discipline you are actually, your domain you are getting to, you need to give the answers or typically project the answers in that particular way. There is no direct access. So if you are working on your own, fantastic. But if it is not, if you are outsourcing something, there is no direct access to the solutions and data scientists. You cannot directly talk face to face, and then you have one-on-one for that matter. And there's a lot of competition going on. You will get from one person something, get another person something. It's difficult unless you understand the subject well. It is difficult to make what is happening out there. And even after having all these things, last but not the least, not necessarily that all the problems data can be able to solve your problems. It could be an open-ended data science problem. And with this, I would like to thank you all for your patient listening. I would be happy to answer.
Yeah, Subhas, Dr. Subhas, thank you for your valuable input about the artificial intelligence in the field of pharmacy. Definitely, your thought might be give input to all of the faculty members belong to the pharmacy fraternity, and definitely they give the path for new inventions and the searching of or hunting of the new drugs. So thank you, Subhas. And again, if some queries is there, then participant can ask to Dr. Subhas Ajmani. just write down the queries in the chat box so we can ask with us Dr. Subhas Ajmani. Yeah, anyone have
Thank
any
you.
query?
My pleasure.
Anyone having any query? Just write down in the chat box. I think it is so.
Either I have gone through or I haven't
lethargy
gone through
type
at
of
all.
things is there because
I'd like to
Kunjibari
touch
sir
base.
is having some queries.
Okay.
Dr. Kunjibari, can you just write down the query so we can ask to Subhas? Definitely no queries comes was there. Okay. Because today there the 5 lectures are there. So the whole
people
day
are already saturated, it
too
seems.
Hectic for all of them.
Yeah.
So definitely.
But any point in time, if anybody has got any query which I can address, I would be definitely very happy to address your queries. You can reach out to me on my email, through WhatsApp, through whatever communication medium you would like to connect with me. I would be happy to connect with you all. And I would just give you best wishes for your upcoming sessions. I hope that it will be fruitful for you, and lot many takeaways will be there for you from this workshop.
Yeah, definitely. Thank you, Dr. Subhas. If anyone have the query, then just send to me and we will forward to the Dr. Subhas, and Dr. Subhas will reply your queries. And now today sessions we will close, and tomorrow we will continue with the new speakers, new resource persons on morning 10:15. So definitely today the faculty members may join on the same link by 10:15 a.m. Thanks to all of you.